Hello. Hello everybody. My name is Dylan Sirodena, and I'd like to welcome everyone to the Cambridge Festival as hosted by the Department of Physiology, Neuroscience, and Development. Today, we have an array of talks spanning these three expansive fields, and it's going to be a really great day for talking about some really cool science. So just a little bit of housekeeping. Feel free to converse in the chat off to the right-hand side. And as well, I'll be keeping an eye on that chat. So if you have any questions there, I will pick them up and I will then be able to ask them at the end of the talk. So without further ado, I'd like to introduce our first speaker, who is Dr. Hugh Matthews, who will be presenting a talk titled The Darkness into Light: Vision, Contrast, and Sensitivity.
Hello, everybody. I'm Dr. Hugh Matthews. I'm a sensory neuroscientist and electrophysiologist working on, among other things, photoreceptor cells within the visual system. And I'm going to be telling you today about how the visual system can operate over a truly enormous range of intensity. Because every day light changes from very dim, imagine you're on a moonless night under a starlit sky, very, very dark conditions indeed, through moonlit nights, early dawn, late dusk, all the way through to very, very intense light under the midday sun. And the visual system has got to cope with this truly enormous range of intensity. And it really is enormous. A white sheet of paper would be about 1 billion times brighter in outdoor sunlight than on a starry, moonless night. But do we actually see this enormous intensity range at one and the same time? Does the visual system really have to cope with this billion-fold or more range at any given moment? And the simple answer to that question is no, it does not. Because most objects do not emit light. Instead, they reflect light from some external light source, such as my light bulb here. So when the light from the light source hits the object, it's reflected. And if it were reflected from the white stripe of my little knitted zebra, the white stripe would reflect a lot of light. And we'd like our visual system to adjudge that stripe as being a white one. But if instead, and we'd say that that white stripe has a high reflectance, it's very good at reflecting light. But if instead a ray of light from the light bulb hits a black stripe, or the zebra's black nose, the black nose will reflect rather little light, and we'd like our visual system to adjudge this as being black because it has a low reflectance. It's very bad at reflecting light. Now, so even though light changes from dim to bright every day, the reflectances of objects in the real world are not going to change. And if you've any sense, you don't spend your time staring at the disk of the midday sun. That would do your retina no good at all. So this enormous brightness range of the illuminating light is translated into a much smaller range of intensities reflected back from the visual world at any given moment. And we can appreciate that by realizing that a white surface only reflects about 20 times more light than a black surface. A very high reflectance white surface, a freshly painted white wall, for example, painted in brilliant white paint, would reflect about 95% of the incident light. If you try really, really hard, you might get up to 98% of the incident light, but that would be pushing it. Whereas a black surface, let us say the surface of black velvet, which not only is not good at reflecting light, but scatters it in all sorts of different directions, might reflect somewhere between 2 and 5% of the incident light. Going below 2% reflectance is very, very difficult. And as a result, the range of intensities we've got to cope with at any given moment is much smaller. And the relative difference between the amount of light reflected from a white surface and that reflected from a black surface won't actually change. It will still be about 20 times as much as is reflected from a black surface, even though the absolute intensity of light reflected changes a lot. So the pattern of light and dark does not depend on how bright the light is, but instead on how good a particular object is at reflecting it. So what vision is looking for is relative differences in brightness between the bright regions, the high reflectance regions of the visual world, and the dark regions, the low reflectance regions in the visual world. And we refer to these relative differences as being visual contrast. Now, I can sense you thinking, but is my visual system really doing that? I thought I was very good at judging how bright things are. I'd like to convince you that that is in fact not the case by demonstrating to you what is known as the simultaneous contrast illusion. This slide shows a stepped background, like a staircase, dark at one end and bright at the other, going up in steps of grey. And superimposed on it is a bar, which appears to ramp smoothly from light grey at one end through to dark grey at the other end. And this is a very powerful percept. The bar appears to ramp. But I'd like to convince you that in fact it's doing no such thing by blocking off the staircase background, the stepped background, so you can only see the bar. And if I do that, maybe a little bit jerky in this zoomed video call, you can see that the bar is actually the same all the way along. It's just a middle grey all the way along. So why did it appear to be brighter at one end, the left-hand end, and darker at the other, the right-hand end? The reason is that your visual system is making a comparison between the bar and the background at each point along its length. At this end, the bar is a lot brighter than the background, so it's being assigned a light grey intensity by your visual system. At the other end, the bar is much darker than the background, and so your visual system decides it must be dark. So we've tricked the visual system by using the mechanism, by confusing the mechanism that it usually uses to extract information about contrast. Let's now get a little bit technical. Let's plot the brightness of the reflected light against the brightness of the illuminating light source, the amount of light reflected from a light or dark bit of the zebra as a function of the brightness of the light bulb. And I'm doing this on what are known as logarithmic axes. A log axis is very clever. Each unit on the log axis represents a tenfold change. So every time we go 1 unit along the log illuminance axis, that's how bright the light bulb is, we make the light 10 times brighter. So 10 times, 100 times, 1,000 times brighter, and so forth. And exactly the same on the y-axis, which plots out the brightness of a particular region on the stripy zebra. Let's plot out the amount of light reflected from a white stripe as a function of the brightness of the light bulb. Every time you make the light bulb 10 times brighter, the amount of light reflected from the white stripe will get 10 times bigger. So we'll move up this thin line as the light becomes brighter, simply reflecting a constant proportion of the incident light. Exactly the same will be true of the black nose of the zebra. The difference is that for any particular light bulb intensity, it will reflect less light. But as we make the light from the light bulb brighter and brighter, the absolute amount of light reflected by both the black regions and the white regions gets proportionately greater. So if we make the light more than 1,000 times brighter, everything will reflect more than 1,000 times more light. And our visual system will have to cope with this brighter range of intensities and make relative judgments within it. To do that, the visual system has got to constantly change its sensitivity. And you can think of this as being like a sort of automatic gain control or automatic volume control for visual sensitivity. And we refer to this process of adjusting the sensitivity of the visual system as light adaptation. And in fact, that's one of the mechanisms that my lab works on within individual photoreceptors within the retina, about which I'll say a little bit more later on in the talk. But for now, let's keep things simple. Let's look at the eye itself and ask how we respond to light. Well, the eye is rather like a little camera. Its optical system is forming an inverted image of the world on the retina. And the retina is rather like the film on the back of the camera, or a modern digital camera, the digital image sensor. We have a photosensitive layer at the very back of the eye containing the photoreceptor cells, which actually respond to light, then layers of neural wiring that process these signals, and finally, the cells that send their nerve fibers along the optic nerve to the brain, so that we can interpret and analyze what we've seen. Now, there are two kinds of photoreceptor cells within the retina. The rods, long elongated rod-like cells, as I say, these are among the cells my own lab works on, and the cones, shorter, stubbier, and as we'll see in a moment, in certain parts of the human retina, very numerous and very closely packed together. And we use different photoreceptors in different ranges of intensity. In the very low intensity range, we use the rods to see the world in black and white. But when the light becomes brighter than twilight or dawn, the cones come into play. And they are used, as we'll see in a little while, in color vision. There's an overlap region in the middle where we're using both rods and cones. But once the rods have run out of steam and saturated so they can't respond anymore, the cones take over, and we see the world in much more detail and in color. But at very low light intensities, only the rods can function, and we see the world in black and white, and as we'll see soon, in rather low resolution. So let's look at how these receptors are distributed over the retina. If you think about it, it doesn't make much sense to refer to the absolute sizes of objects and images on the retina, because as an object in the real world, it moves further away. It appears to get smaller and smaller. It makes much more sense to follow the trajectory, the path followed by a ray of light from some particular point of the object, and look where that particular point on the object is imaged on the retina, and to express that as an angle of displacement away from the very central region of the human retina called the fovea. So in this example, I'd be looking, let's say, at, let's flip our candle upside down. That would be another, that would be something very, very much at the bottom of the candle. It will form an image of that particular point on the candle at a position displaced in the opposite direction on the retina, in this particular case at a visual angle of 30 degrees away from the fovea. Using that scale of angle, visual angle, let's look at how the rods and cones are distributed across the human retina, starting off at the very center, the fovea. In the fovea, in the human, there are very, very few rods, but extremely densely packed cones. And as we'll see in a moment, this allows central human vision to be very detailed indeed. But as we move away from the fovea, the density of cones falls away precipitously, and the density of rods rises. So at about that point there, we've got rather sparsely packed, bigger cones, and little rods, the purple ones, packed in between them. And if we move to the periphery of the retina, the cones are very, very sparse indeed, and most of the receptors we see will be rods. So why does the high cone density in the fovea matter? Well, to resolve detail in bright light, we've got to be able to see the difference between parts of the image that stimulate cones and parts of the image that don't. So to see this letter E, we would need an unstimulated cone in between each stimulated cone so that we could see the dark gap between the two red prongs. If the E were smaller, we wouldn't be able to see that it was a letter E because we wouldn't actually reliably see the gaps. We'd simply see a blurry red blob. This would not be resolved. So to resolve fine detail, we need to point the very tightly packed cones of the human fovea at that point of interest on the object. And we also have to have bright enough light that the cones can function. But when the light is very dim, we use our rod photoreceptors instead. And therefore, if you want to see something very dim, For example, if you want to see a dim star in the dark night sky, it'd be a good idea to look to one side of it so that you point your cone dense fovea away from the object you're interested in and allow the image of the star to fall on your very densely packed rods in this region a little bit to one side or the other of the fovea. Now, let's contrast the human with the cat. Cats hunt at night. My cat certainly hunts at night and occasionally brings little offerings back to place on the doorstep for the next morning. And the cat retina mostly uses rods. There are an enormous number of rods in the cat retina, even in its very central region, position 1, and rather few cones. Perhaps even in this central region, 10 rods for each cone. And as we move more peripherally and the cone density falls away, the ratio of rods per cone becomes even larger, ever larger. And that means that the cat can see very well in the dark because it has highly sensitive rods at all locations within its retina. But what is the nature of the image that rods will see? Well, light is not continuous, but it's instead composed of particles of light or photons. So a dim, steady light is a stream of single photons arriving randomly at some mean rate that would correspond to the brightness of that particular point on the image. And to detect the image, we've got to actually detect the photons. I can sense you thinking, why is this a problem? Let's imagine how we're going to detect the individual photons, how we can make it a reliable process. Well, you saw in my earlier scanning electron micrograph, the picture of the rods and the cones, that rods have very long photosensitive parts of their cells called the outer segments. And within these outer segments, there's a stack of membranous discs, rather like a stack of pennies inside the cell. And the molecule that actually absorbs light, a molecule called rhodopsin, the photopigment, is contained within these membranous discs. And these discs are stacked up along the rod like a stack of pennies. And because we've got perhaps 1,000 discs within each outer segment, we have a very large number of rhodopsin molecules stacked up within those discs. And therefore, this large number of pigment molecules makes it likely that as light passes along the length of the outer segment, the photon will in fact be absorbed. It's not a certainty, but about 2/3 of the time it will nonetheless get absorbed if it's of the right wavelength, a sort of blue-green wavelength, very close in fact to what's called Cambridge blue. And rods actually can respond to individual single photons. This is a picture from my own lab of an individual salamander, a rod photoreceptor, whose electrical responses are being recorded by this glass pipette, allowing the recording of the responses to single photons. Let's imagine we present very dim flashes of light. Every now and then, rhodopsin catches a photon, and there's a little response. But sometimes the photon makes it to the far end of the outer segment without getting absorbed. There we had a failure, there another failure, there a success, there another success. And here I think we got incredibly lucky. 2 photons were absorbed at the same time from the same flash, and we got a bigger response. So individual rods can indeed respond to single photons. These dim flashes can evoke single photon responses. And the same will be happening when the rods within our own retinae respond to very, very dim light. So what will be the consequence for the image that we see when light is very dim? Well, when the light's very dim indeed, we will be catching so few photons that the image will be very noisy. I would challenge you from this image produced way back in the 1950s using a very high sensitivity electronic camera, not digital in those days, in fact, too long ago. I would challenge you to tell from this image showing the single photons arriving and being caught by the camera, what this is actually an image of. So it's rather like the pattering of raindrops on a roof, the arrival of single photons. When the rate of falling of raindrops is very low, the density, where you've got more or where you've got fewer hitting in each given area, is really not plain. You can see there's something there in the middle, but what it is very unclear. But if I make the light a little brighter, you start seeing a ghostly face. It's somebody's face. If we make it a little bit brighter still, you can definitely see it's somebody's face. And a bit brighter still, it appears to be a female face. Certainly longer hair. Now we can see more detail. Now we're moving probably into the range where your cones would start responding and giving you more detail still. And so, as the light becomes brighter and brighter, the noisiness of the image diminishes. Now I can sense you thinking, but why can't we see in detail even at low intensities with our tightly packed rod photoreceptors? Because there were a lot of rods per unit area, both in the human and the cat. Won't rods let us see detail after all? Well, the answer is, as you've just seen, that there isn't detail there to be obtained. The rate of arrival of photons is so low that we can't really use the fine grain of the array of rods. Instead, we want to add the signals from our rod photoreceptors together to make a bigger bucket to catch photons in. And that's exactly what the retina does. It adds together the signals from many rod photoreceptors to send the signals from perhaps some 1,500 photoreceptors out along a single optic nerve fiber. So the probability of one of these rods catching a photon is reasonably good. And so the optic nerve can signal along that particular fiber, I've seen something. But the detail of exactly what it was isn't resolvable on a fine spatial grain. So we've smoothed out the noise, but we've destroyed our ability to use the rods to see fine detail. So what does determine the dimmest light that you can see? Well, to work this out, let's not use a human, but instead a toad. And we use a toad for the following reason. Heat can excite rhodopsin too, but much, much less well than light. And amphibia, including toads, are cold-blooded, so we can change their temperature, their environmental temperature, and therefore change the rate at which individual rhodopsin molecules in the rods will be excited by heat. And the toad is in near darkness, minding its own business. It's presented in these very dim light conditions with a white dummy worm underneath a perspex screen. And toads, when they see what appears to be a worm wiggling, strike at it with their tongue to try and eat it. And if you look for the intensity at which the toad strikes at the dummy worm with its tongue, it means that it can then see the dummy worm. Let's plot out that absolute threshold at which the toad will strike at the dummy worm. And against it, again, these are on log coordinates. Note each unit, each big tick gets 10 times bigger. And plot against that the rate at which we would expect rhodopsin to be spontaneously excited by heat. As the eye gets warmer, the rhodopsin is excited more frequently by heat. And even more noise is therefore added to the background, above which we've got to detect that there's actually something there. The more noise, the harder this becomes, so the more light and the more photon absorptions we need to do that. And that's exactly what you see happening here. As the temperature increases, the absolute threshold progressively increases, and the rate at which rhodopsin is excited increases, and they increase in tandem. Every time you make the toad sufficiently much warmer to increase the absolute threshold by a factor of 10, it does so because the rate of isomerization increases by a factor of 10 also. And humans, at normal human body temperature, are much worse at this than toads because we're warmer, and therefore rhodopsin is being excited by heat at a higher rate, even in complete darkness. How can we do better? How can a cat do better than a human? Well, any cat owners or anyone who's seen a cat on a dark night when the car is driving towards the cat will know that a cat's eyes reflect light. And the reason they do so is that they've got a shiny layer at the back of their retina called the tapetum lucidum, which in Latin literally means shiny carpet. And the shiny carpet, or tapetum lucidum, reflects light back again through the retina. Why might this be a useful thing to do? What it means is that light is going to pass through the retina twice over. The photon might get absorbed by the photoreceptor on the first pass through the retina, but if it doesn't, it'll get reflected back again and get a second go. So this allows the cat to try and use every available photon in its very numerous rods, and thereby increase its sensitivity. The problem for the cat is that if you absorb light here, it would appear to be from that point on the visual image. And if you absorb it there, it would appear to be from that point on the retinal image. So this process of a double pass of light through the retina, the reflection of light, makes the image more blurry than it otherwise would be. But the cat's using its rods where the image is already rather low resolution, so it doesn't care. Arctic reindeer have a rather clever trick. They change the color of their tapetum between winter and summer. If you were to look deep into the eyes of an Arctic reindeer in summer, you would see a beautiful golden green color. But in winter, a much lower reflectance, deep blue. Very little light is reflected in winter, and it is of a shorter wavelength, blue rather than golden yellow. So both less light is reflected out and of a different wavelength. Why might that be useful? Well, if less light is reflected out in winter, more light is scattered within the retina and therefore may get absorbed. Whereas in the summer, reflected light passing out is simply lost. So this scattering of light within the winter eye makes the reindeer more sensitive in winter than it would be in summer. And the mechanism it uses to change the color of its tapetum is almost pathological, really. In the winter, the extraocular pressure within the eyeball increases, almost blowing it up a little bit like a little balloon. That in a human would be called glaucoma, and if it were to persist for too long, would actually kill the photoreceptors and other cells in the retina and lead to blindness. But in the reindeer, this mild glaucoma squashes the fibers of the tapetum, which reflect light together, so that they are closer together and reflect and scatter light at shorter wavelengths and scatter light more effectively. So a sort of seasonal glaucoma, mild seasonal glaucoma, lets the reindeer see with greater sensitivity in the winter than in the summer. Now, to see the world in colour, we use our less sensitive cones. And human vision responds to sunlight, which contains all the colours of the rainbow. We've evolved to respond to this range of wavelengths from blue-violet at one end of the spectrum, through blue, through green, through yellow, then orange and red. And our cones have got to sample these different wavelengths to provide us with a reliable image of what we have actually seen. Let's go back to our little stripy zebra, but this time a colourful zebra, and again illuminated from an external light source. If a particular stripe, let's say this stripe here, is good at reflecting short wavelengths of light, blue, we would like the visual system to say, I've seen a blue stripe. If instead it reflects a long wavelength of light, or red, we'd like our visual system to say, that's a red stripe. Now, why is that a problem? Well, it's a problem because vision has got to try to work out what colours something can reflect. That's known as the object's spectral reflectance. irrespective of the color of the illuminating light. And in the real world, illuminating light changes quite a lot. So we'd like our visual system to be able to judge colors, whatever happens to the illuminating light. We'd refer to that constancy as color constancy. But light from different sources varies tremendously. Average daylight is quite well behaved, plotting out the relative intensity as a function of the wavelength. But artificial light can be really weird. This is the spectrum fluorescent tube, and it's got spikes in the violet, the blue, the green, and the greeny yellow, and then this fuzziness in between. And we'd like our visual system to be able to judge colors, irrespective of whether we're looking at the objects under daylight or fluorescent light or tungsten light or something else. So how can we judge spectral reflectance reliably? The answer is by looking for differences in color. And this is known as the process of color contrast. In this example, the red pepper pops out from its background of green Granny Smith apples really very obviously. You can immediately see there's a red pepper there. You don't have to search for it. How do we do this? Well, humans have three different kinds of cone photoreceptor, responding to blue, green, and red wavelengths. So roughly speaking, we call them blue sensitive, green sensitive, and red sensitive cones. The blue sensitive cones respond to the bluey violet wavelengths, the green to the yellowy greeny, and the red, slightly longer, it's doing better in the reddy orangey region of the spectrum. So how might we use these three classes of cone when looking at a colourful object? Let's use as an example an apple tree with green leaves and red apples. The green leaves will, unsurprisingly, be very good at reflecting blue-green wavelengths, especially these greeny ones in the middle of the spectrum, whereas the red apples will be reflecting the longer wavelengths much better. And as a result, when we look at what is present using each individual type of cone, we find that in response to the green leaves, our green-sensitive cones are responding very well. And when looking at the red apples, our red-sensitive cones are responding very well. And it's by using the balance between these different cone responses that the brain can decide what colour an object actually is, and whether or not it's different from its background. So, human beings have three cones. We're therefore said to be trichromatic. And this trichromacy means that if we pick reasonably sensible primary colours, let's say blue, green, and red, we can blend these together in appropriate proportions to match a test light. And this, of course, is the principle being used by the display screen you're looking at the moment. The display screen is mixing red, green, and blue light to present the perception of different colours by appropriately stimulating your different cone classes. How well a particular display screen does this will determine how accurate its representation of what you might really see in real life will be. So why is it useful to humans and other old world primates to be trichromatic? Well, without separate green and red cones, you cannot see fresh leaves and ripe fruit. So when, some tens of billions of years ago, old world primates produced, if you like, a mutant version of what was previously a yellowish pigment, so thereby producing red and green separate cone pigments, this was quite a useful survival feature, and so was evolutionarily retained by natural selection. In contrast to primates, which may have three different cones, most mammals, dogs and cats and farm animals, have just two, blue sensitive and greeny yellow sensitive. So a human can very easily tell a Braeburn apple from a Granny Smith apple. The Braeburn apple is nice and red and rosy, and the Granny Smith looks green. Perfectly tasty, but it's green. But to a horse, both of them would look golden yellow, just like a russet apple. I think the horse will probably accept either of them, so that will be just fine for the horse. But if you are a canopy-dwelling primate searching for ripe fruit, being able to tell red from green is clearly a plus. Many birds, in contrast, are tetrachromats. Tetra meaning they have 4 cone classes, and they have an extra pigment in the ultraviolet, allowing them to see even richer colours than we can. So colour vision is looking for differences in colour, and there are 2 different ways in which it might do so. It can look for differences within an object. Is a particular region a bit greener or a bit redder, or a bit yellower or a bit bluer? We would call this colour opponency within an object. Alternatively, it can look across the boundaries of an object, between the object and its background, such as the situation with my red pepper on the bed of green Granny Smith apples. Is the object redder or greener than its background, or yellower or bluer than its background? This is referred to as colour contrast. And I'd like to convince you over the next few slides that your visual systems really are extracting these different sorts of information, starting with colour opponency. This illusion is a rather nice demonstration created by the Japanese psychophysicist Akiyoshi Kitaoka, and it demonstrates the process of colour opponency. I'd like you to look very steadily at the little black cross in the middle of this slide, and keep doing so for the next 10 or 15 seconds. Just stare at the black cross, don't look away from it. Then I'm going to move 1 slide forwards, and I'd like you to keep looking at the black cross, don't look away from it. And what you should then see is a series of dots appearing of the opposing colours, the contrasting colours in the white region in between. And the reason that this happened when I jumped the columns of coloured dots to one side is that the cells signalling different colours within each individual little circle had adapted and stopped responding to that particular colour. So when I presented white, which stimulates all cone classes equally, this looked like the opponent colour, because we'd upset the balance between the different colour signals. Let's now look at an example of colour contrast. In this slide, and again, the palette of colours I've used was selected by Akiyoshi Kitaoka, it shows a pink square on the left side, a big pink square, and a big green square on the right side. And superimposed on these big squares are little squares of approximately the same grey. Maybe little differences on your screen, but they should look approximately the same. Actually, that's a complete illusion, as I can show by sliding the squares, little squares, sideways. And you can see, sorry about the jerkiness, this is the problem with Zoom, you can see that they're actually extremely different. So why did they originally appear the same? And why do they now look even more different than they really are when superimposed on the other background? Well, this is an example of colour contrast. The red background makes the square look greener, and the green background makes the square look redder. And this can be used to practical effect. If any of you have ever visited Walt Disney World in Florida, in the Epcot Center, the foliage and the grass looks wonderfully green. And there's a visual reason for that. Walt Disney commissioned Kodak in the 1950s to choose the shade of the pink pavement to literally make the grass look greener through color contrast. The grass really does look greener at Disney World. Now, we need differences to tell what color things are. And we need to do this all the way through the normal day because with an external light source such as the sun, some objects will be in direct sunlight and some will be in shade. And also, the illuminating light changes in color as the sun rises or sets. Just after sunrise or just before sunset, we have the golden hour. Everything looks very golden and very wonderful. Amateur photographers make use of that to make landscapes look extremely beautiful. And just before the sun rises, or just after the sun sets, we have the blue hour, when scattered light from the sky makes everything look blue. And we've still got to be able to judge relative colors as best we can. Even in the middle of the day, direct light from the sun is a bit yellower, And in the shadows, there's more scattered blue light from the sky. So the balance between yellow and blue changes, between direct sunlight and shade. And that can in fact be useful. Objects are dimmer and more blue in the shadows. And that is a good way of telling that you are seeing a shadow rather than a real edge. So your visual system is trying to compensate for sun and shade to find out what objects in those different regions of the image are really capable of reflecting. Let's try and trick this system. Comparisons between these different what's happening in each region normally let us see colors as constant. For example, you might say in this region of direct sunlight, those green leaves are very good at reflecting middle wavelengths compared to the things around them. They therefore must be green. Even in the shadows, we're comparing objects with things immediately next to them. Let's try and trick this system with a synthetic image by using a cartoon created again by my favorite psychophysicist, Japanese psychophysicist Akiyoshi Kitaoka, showing a cartoon of a little Japanese girl, half of whose face is illuminated with white light and half of whose face is in a shadow illuminated with blue light. And she's got a yellow hair bead in her hair and that's odd. She's got one yellow eye and one grey eye. The yellow eye, in fact, looks remarkably like the yellow hair bead. But is it really so? Let's find out by blocking out the blue on the left-hand side of the image. And if I do this, if I slide down a little mask blocking out the blue background, you can see that the two eyes are actually exactly the same. So why did the eye on the blue side appear to be yellow? The answer is our visual system was doing the best it could to judge what color the eye must be, based on the assumption that that half of her face really was illuminated with blue light. But the eye wasn't reflecting enough blue light. Therefore, it must have been yellow, thought our visual system. This is meant to help you see the real colors in a real shadow. But by using this synthetic image, we've tricked the visual system, confused it with this simulated image to create an illusion. Now, at sunset, scattered light from the sun is very red, and so red, in fact, that we haven't got a wide enough range of colors to make a proper judgment for color constancy. So at that point, really at the end of the golden hour, color constancy tends to fail, and colors do look wonderfully golden. Try it the next time you have a nice red sunset, and you'll find the world is a very golden and happy-looking place. And what we expect the lighting can govern what we actually perceive. A famous internet meme of a few years ago is the meme of the dress, which some people saw as being gold and white. It in fact was a gold and white dress, but some people perceived as being a black and blue dress. Why might that be? Well, psychological experiments have shown that expectation very much governs whether you see it as gold and white or black and blue. If you're an early riser, you're assuming blue-tinted light, and you see it as white and gold. But if you are a night owl, used to either sunset or yellowish artificial light, you see blue-black. You're assuming yellow light. So the majority, in fact, in that particular psychological experiment fell into the yellow light category, the night owl category. Now, you may be thinking, no, I couldn't be fooled like that, could I? Well, actually, you could. Here's a rather nice synthetic image designed by Kasuga, showing a cartoon of this dress image. And again, splitting the image into a shadowed and a direct section. On the left-hand side, black and blue. On the right-hand side, gold and white. But you can see from these little pipes running from one side to the other that the dress and the apron are exactly the same on both sides in the shadowed region. But a very powerful expectation has been generated on each side that this is a non-uniformly illuminated figure. And try as you might, it is almost impossible to make the percept shift from what you've been led to believe. So your visual system is capable of tricking you quite considerably. The mechanisms that we use under normal circumstances to try and see the world as it really is can sometimes be fooled to make you see something quite different. And on that note, let me finish with a final slide that appears to show a very bright light shining from the middle of the screen from among these petal-like segments. So is the screen really brighter in the middle? Could it be brighter in the middle? Let's find out by blanking off the petals. A little jerky again, but you can see that actually the screen in the middle is exactly the same. And we've been fooled into thinking that it's different through the progressive gradient of brightness along each petal, which makes us think that the light must be getting brighter behind them as well. Only it isn't, it's exactly the same. So I hope I've convinced you that light and dark are important, that the visual system has some very clever mechanisms within itself to sort out this enormous range of intensity, and that these mechanisms can be tricked to produce a number of quite dramatic illusions. Thank you for your attention.
Thank you very much for a very illuminating talk. Now, I'm looking towards the chat right now. For those who are watching, please feel free to ask any questions there, and I can communicate them to our speaker. I have a couple myself. One in particular, you were talking about the various cones and how they receive the various wavelengths of light. How does that relate to color blindness? Oh, I think we may have lost our speaker, which is very sad.
Yes, I believe so. Let's see if we can log in again shortly. Bear with us.
Well, hopefully we can get him back. However, if we cannot, we'll be having our next speaker, Connor Ross, starting around noon. We'll be talking about wind signaling in particular.
Sorry, Dylan, Zoom crashed on me there.
Oh, excellent. I'm glad you could make it back. Can you hear me all right?
I can hear you. Can you hear me?
Yes, I can.
Then
Excellent.
good to go.
I'm not quite sure if you caught my question, but I was asking about color blindness and how it relates to cones.
Color blindness is great fun. Had time permitted, I could have said a lot about color blindness. As I said earlier, in evolutionary terms, the red and the green pigments in humans are relatively new inventions. Tens of millions of years ago, but in evolutionary time, it's the blink of an eye. And they're very close together on the human X chromosome. What that means is mistakes can occur. What sorts of mistakes might those be? Well, when creating a gamete in meiosis, you are crossing over chromosomes, you're selecting what you want to send on to the gamete. And the problem is you can either delete one of these red or green pigments, producing an X chromosome that only has red or only has green, or perhaps even more confusingly, produce a hybrid pigment instead of a red or a green, one that's sort of half and half. The result of that half and halfness is a pigment that would respond to the yellow. Now, a human genetic male has just one X chromosome. If you inherit an X chromosome that has a missing cone pigment, you will not be able to respond differently to that particular wavelength, range of wavelengths. You might lack a red cone pigment. You would therefore only be using your green cones to look at that medium to longish range of the spectrum. So you wouldn't really be able to tell red from green. That person would be red-green colorblind. If you were to inherit a hybrid pigment, let's say a half and half, instead of either your red or your green pigment, you would see the world as a trichromat, but you'd be different from the usual trichromat. You would be a so-called anomalous trichromat. Now, you might be thinking, so is there such a thing as a color normal human? Actually, no. There are quite common polymorphisms that have very, very subtle effects on exactly where the red pigment, which range of wavelengths the red pigment responds to. So in that sense, there's no such thing as a precisely color normal. In some other species, it gets even stranger.
Interesting. I had another question also about kind of on the dim light part, where you mentioned that oftentimes in humans, when it's dim light, more and more rods are being used. Is there a sort of buffering system where the brain kind of waits for more information in order to collect something to make an image?
You're in a sense buffering right out at the sharp end, at the receptor itself. Rods respond really rather slowly, especially when the light is dim. You're collecting together light over what's called the receptor's integration time. So in a sense, you're buffering up in the receptor itself. Have I seen a photon recently? Have I seen a photon recently? When the light's really dim, that integration time is quite long. In an amphibian, you're talking about a second or so. They really are that slow at really dim intensities. Humans, it's not as bad, but you can track this and also the speed of the rest of the visual pathway through the retina all the way up to the center by using what's known as the flicker fusion frequency. That is the frequency at which a flickering light appears to be steady. And you might think, so is that really a thing? Well, lots of light sources, artificial light sources around you are flickering all the time, but they're flickering at 100 times a second, double the mains frequency, and that's above the flicker fusion frequency even of the cones. It's too fast for the cones to see as a flicker. The rod system is more sluggish. You can cheat it and make it think, even when the light's quite bright for a rod, that the flickering light at 20 flickers a second is steady. Cones can see that as flickering. The rods say, as far as we're concerned, it's steady. And go dim, the rods get even more sluggish.
Interesting. Well, I do not see any more questions in the chat. So then I'll take this opportunity to thank you, Dr. Matthews, for coming and, of course, giving a really great talk. So we will then have a very short break, and in about 5 or so minutes, we'll be starting our next talk. So we'll see you all then. Hello and welcome back. We're going to be starting our next talk now, who is going to be Connor Ross, who'll be presenting to us today on Wind signaling, a Pandora's box. Connor, I'll let you take it away.
Brilliant. Thanks. Cheers, Dylan. And thank you to the organisers for giving me the opportunity today to talk to you about a little bit about some of my research, but mainly a background to my passions and research in this particular area. So I'm by default a developmental biologist. I study both embryos and stem cells. I've been really interested in this ever since I was quite a young boy, really. And my main aspects of research involve trying to understand how cells communicate to each other, with a particular focus on one mode of ancient communication, which is wind signaling, which I'll talk about today. So to give kind of a background to this, if my slides decided they actually want to move. There we go. So the overview of the talk is I'm going to quickly introduce development of the embryo in humans very briefly, and stem cells, what they are and how we obtain them, and then move into talk a little bit about the history of the wind signaling pathway, what we've learned from studying it in embryos and stem cells and its overarching implications in human disease and development, and then looking into a little bit more detail of how cells coordinate fate specification events, but also more basic, looking at how wind signaling goes wrong and how it underpins diseases. So to kind of overview this, human development is rather quite astounding, and we live in an age now where we can actually study this a lot more focused, and it's really, really cool. Basically, the gist of it is that upon fertilization, within the first 5 days, the early embryo is first of all making its way through the fallopian tube towards the uterus, and is undergoing a series of divisions that allow it to basically grow in size in preparation for implantation into the uterus. And around about day 5, the embryo undergoes a stage of compactation, and it forms something what we call the morula. And then over the next 24 hours, it expands to form a blastocyst, which is shown here. And at this stage, there are two main populations of cells. The first one of these pink cells here, these are they form the ICM, the inner cell mass, and these contain the progenitors of the epiblast and the hypoblast, which are the two lineages that give rise to the embryo, the fetus, and then the underlying cells give rise to the yolk sac, which nourishes the embryo during early development. And the other cells around the outside, these are called trophoblast, and these cells are precursors of the placenta. So the embryo, prior to implantation, has the cells necessary to proceed, and then on day 7, the embryo begins to adhere to the uterine lining of the mother and begins to embed and implant, ready for the next stages of development. So that's what the cartoon looks like. This is what it looks like in real life. And these are IVF embryos. So you can see that during the first 5 days up until day 5, 5.5, and then we get formation of this blastocyst, and then where this white arrow is pointing to demarcates this inner cell mass, which I showed you in the previous slide. And in here are the cells that go on to form the embryo, which is shown here. And this is where we get embryonic stem cells from. And then on day 7, the embryo literally hatches from this shell. Very similar, is analogous to how a chicken will hatch from an egg, or anything that hatches from an egg. Basically, this is an analogous thing. There's still this eggshell around the outside called the zona pellucida. The embryo then hatches and then attaches and implants. And this is a really nice image of around about an 8 or 9 week embryo / fetus, where at this stage you can see that it has the eye, it has the beginning of an ear, and it has the limbs and then beginning of the fingers. And so at this stage, the embryo / fetus has all of its organs. And then from the 9th week onwards, when it is demarcated as a fetus, the rest of gestation, at least in humans, is dedicated solely to growth and the preparation for birth. And after birth is when a lot of changes happen. So one really important question is, well, people would have generally heard is, what is a stem cell? We've seen it in the media quite recently, and they're really quite exciting, and they hold quite a lot of promise, which I'll describe in the next slide. But basically, a stem cell in its purest form is a cell that can self-renew or divide for long periods of time and remain as an undifferentiated stem cell. They don't have any kind of fate. They have their fate bestowed on them via cell signaling pathways and their ability to communicate between the cells. And the other feature of this is that in doing so, they can differentiate. So we can take a stem cell in the lab, and we can push it and make it form liver cells, heart cells, cells of the eye, cells of the inner ear, cells of the teeth. You name it, we can pretty much do this. And the other quite important feature of stem cells is that whilst in their undifferentiated state, they remain competent to receive signals to then allow them to differentiate. And so in this diagram, you can see that they do one or two things, which we term as either symmetric or asymmetric division. So in a symmetric division, they basically form two daughter cells of themselves, basically. And then you can have an asymmetric division where you have the stem cell will continuously divide in its undifferentiated state, it doesn't have a fate, and then eventually a cell comes and it has a fate. It begins to differentiate into a certain kind of cell. So because of this and the fact that we can culture these stem cells for long periods of time and they can form lots of other different cells, they hold great promise for regenerative medicine and basically to study human development outside of the embryo that would be otherwise inaccessible. And some of these things that we can use stem cells for is to understand how birth defects occur within the DNA. We can create all these different cell types, as I kind of highlighted previously, for transplantation in the future. We're kind of just starting to step into that window now. And then we primarily use the stem cells as a tool to look at drugs and understand how drugs interact with tissues, and understand how we can tailor drugs to specific individuals. So the stem cells hold a lot of promise, but there's a lot of things that we're still quite far away from doing. Like I say, transplantation, we're looking more into it. Scientists who work in this field more specifically are looking into this, but there's still a lot of things that realistically need to be understood about these cells before we can start, kind of pumping them into people really. So we know what the stem cells are, and we know that they can differentiate and they remain as stem cells in the lab. And there's a rich history to the background of stem cells, and this is a nice diagram and a roadmap to it. And there are several different types of stem cells that exist. The original stem cells that were kind of well characterized back in the day, in somewhat of a basic form, if you kind of look back compared to what it is now, were stem cells derived from the embryo. And in the 1980s, this was first done in the mouse by Martin Evans and Matthew Kaufman, and they were able to derive embryonic stem cells straight from mouse embryos and culture them for long periods of time. And then it wasn't until 1998 when James Thomson's laboratory were able to derive cells of a slightly later stage from the human embryo, which was quite a massive feat for the stem cell field. And then even more so and more recently, the work from Shinya Yamanaka in his laboratory has demonstrated that actually what we can do now is we can take skin cells, fibroblasts, and we can effectively reprogram them and switch their fate to then turn them back to pluripotent stem cells, which are termed induced pluripotent stem cells. And these can be then differentiated into, as I say, all these different types of tissues, and then they can be used as treatments for therapy, and then they can also be used to screen drugs. And they, like I say, they hold a lot of promise. The other type of stem cell that exists within adult tissue, and this is something I'll describe a little bit later on in more detail, are these multipotent adult stem cells. So, for example, your bone marrow contains these multipotent stem cells, which allows them to differentiate into the blood. And basically, the whole point of these stem cells in adult tissues, these niche stem cells, we call them, is the fact that they play a primary role in regeneration of blood, the intestines and things like that. And this is what I'll come back to. So one term that I threw around quite a bit there was pluripotency. And this is the term that we associate to these stem cells based on their capacity or ability to differentiate into cells. So pluripotency is defined as the term that they can form all types of cells. Multipotent stem cells can only form a few types of cells. They're generally very restricted to what they can do. But if we fast forward time and move through the decades, a lot of work has been done to try to understand at least mouse embryonic stem cells in a lot more detail. And the reason for this is that the original conditions which these stem cells were generated in utilized fetal bovine serum, which is a blood component from bovine fetuses, which was spun down and then used. And they were all the stem cells were cultured originally on inactivated skin cells from the mouse. And basically together, they maintain stem cells in their pluripotent form is what I said. But the problem then came to that the fetal bovine serum that was used was quite variable, and it contained a lot of undefined growth factors. So it supported the stem cells, but we didn't know what it was doing. And then as we move through the time, a lot of fantastic work from Jenny and Austin here. To note, this is Jenny, my supervisor, reformulated a lot of the conditions, and gradually over time, they moved away from using the inactivated skin cells and eventually moved away from the FBS, even though the conditions are still used widely today. And then in 2008, 2009, Austin's postdoc at the time, Keelong Ying, generated a completely defined culture medium for these embryonic stem cells. And then this is what we come to really. So the process originally was that you would take an embryo, a mouse embryo, and you plate it down. And then this blob here in the middle is the inner cell mass, what I described before. And then eventually it grows. And then Jenny did the majority of this kind of work is you were able to take this, this inner cell mass outgrowth and you would dissociate it with it very gently. And then eventually you would start to get these little baby colonies arising, which is, again, obviously really, really cool. And then if we fast forward time again to 2015 to 2016, this is the story around about from this time and progressing onwards. Jenny and help from Gugwa, they were able to generate and derive embryonic stem cells of a similar stage in the mouse from human embryos. So if we look through this is a day 6, day 7 human embryo. And what Jenny has done is remove the outer cell layers of the embryo, which is here. And then what you're left with is this little tiny ball in the center. Remember, these contain the cells that will give rise to the embryo. And then what Jenny has done is she's basically dissociated all the cells and plated them out. And then what you get is individual cell colonies, which we can keep in the lab and keep them growing for very, very long periods of time to study and give us ideas of what actually happens during the early stages of human embryonic development. So to summarize what I've told you so far, and to kind of keep everybody in the loop, the stem cells remain in their undifferentiated state. At least embryonic stem cells with this pluripotency can form all the cell types of the embryo. We derive embryonic stem cells from embryos. We can do it in mouse, we can do it in human, we can also do it in a lot of other different species as well. The early stage human stem cells are derived from IVF, surplus IVF embryos, but what we can also do now is derive the induced pluripotent stem cells, as I mentioned before, from skin cells. And naturally, because of this, stem cells hold a great amount of promise for regenerative medicine and in our purposes to study human development and to treat diseases. So now I want to move a little bit on to understanding and demonstrating what the Wnt pathway is and how it regulates as a form of cell communication. But before I kind of do that, I thought it would be a nice idea just to very quickly run through the anatomy of a cell because it's quite important for the next part of the talk. So this is a very widely accepted image of the cell, and for the last 50 years or so, there's been minor tweaks to it. We've developed our understanding of the cell and generally, a lot of what you see here hasn't really changed a whole lot, apart from the more finer details. But the main parts I would really kind of like to draw your attention to is the outside of the cell has this orange, somewhat of a barrier, really, but it's not per se a barrier. This is what we call the plasma membrane, and this kind of separates the inside of the cell and all of its kind of constituents from the environment, and the outside is what we call the extracellular environment. And this purpley blotch here is the nucleus, and inside the nucleus contains what we call chromatin, or in smaller forms, it's the DNA, basically. The DNA is the blueprints of the cell, it's the blueprints of any organism. And the really important part about this is that in your DNA, which is kind of condensed onto 46 chromosomes, so you have 23 over 2 pairs, you get 1/2 from mum, 1/2 from dad. And what we have is the DNA, and then there are long tracts of DNA, which are regions for what we call genes. And these genes code for proteins, and the proteins are the effectors of the cell. And they basically tell the cell what to do, is kind of the bottom short of it, really. So in terms of the Wnt pathway, which I'll go on the next slide to describe, it's a form of signaling. And this is a method by which cells communicate with each other. So what cells will effectively do is they create these proteins and they push them out into the environment, the surrounding the cells. The signaling molecules, also known as ligands, bind to receptors, which are more proteins on the surface of the cell. And then we get a transduction cascade through the cell, and then it activates a response, generally within the nucleus. Now, not all receptors are found on the surface. There are examples of some receptors, mainly receptors that transduce the sex hormones. They're actually found on the inside of the cell. But generally, what these are doing is they are communicating signals from outside of the cell to the inside of the cell. That's the main kind of branch of it. So the Wnt pathway, as I mentioned briefly before, is something I'm very, very interested in, and it's evolutionarily conserved all the way back around about 380 million years ago. So if we go back 380 million years ago to very somewhat simplistic organism compared to modern day organisms, you find the rudimentary aspects of Wnt signaling still exists. So what's changed is that throughout evolutionary time, things have got a lot more complex. And a lot of the work that was originally done on the Wnt pathway, at least in this initial discovery, was done by Roland Noosa, and at the time was his supervisor and then collaborator, Harold Varmus. And the discovery of the Wnt protein comes from 2 separate lines of evidence, and they both merged at the same time. So Roland Noosa, during his very, very early work as a PhD and early postdoc student, was very interested in understanding how breast cancer forms via mammary tumors in the mouse. And basically what he showed was that a retrovirus, MMTV, the member of the family, was involved in causing these mutations in mammary cells. And basically what happens is you get the formation of cancer. And then when he went away and sequenced the genome and looked closer, he actually found that this INT1 locus was responsible for it. And it's mutated in mammary tumors, and basically the protein codes for the actual ligand that binds to the receptor. But because it was so highly expressed in these cells, he found that actually it was involved in development of the mammary tumors. At the same time, there was work done in the Drosophila that if you remove the other protein that's found in fruit flies, DINT1, you actually get fruit flies that are wingless. They don't have any wings at all. And then it was later shown that these form morphogen gradients that are involved in patterning the fruit fly. And so what actually happened was that these 2 discoveries came together. And so INT and then the wingless, which is what it was called in the Drosophila, they were found to be homologous of each other. So they're the same proteins, but they just have slightly different functions. And then the word Wnt was born. And then from there, the whole field exploded, really. And it was actually, from what I've read of accounts from both Rola and Harold, the field was just exploded, really, and it was fantastic. And then a lot of work went into mapping the pathway, which I'll describe in the next few slides. And then if we come to more modern times, a lot of focus has been pushed onto trying to purify these proteins because they really actually don't like to be outside of the cell for too long, and it makes it quite difficult to discover, to work with, sorry, shall I say. But in the 1970s, around about the same time as Roland Nusse and Harold Varmus were working on the Wnt pathway, Eric and Christine both made a very interesting discovery that actually a downstream protein of the Wnt pathway called, in fruit flies, armadillo, and in humans, beta-catenin, and other mammals, was involved in the pathway. And this excerpt from Eric is quite nice, actually. So the name comes from a series of Wnt screening experiments that were done in the 1970s, which generated fruit fly larvae with mutant cuticles. The cuticles are just part of the fruit fly. You can actually see them here, and then the bands and the mutants. And basically, when you remove the wingless protein, the armadillo, my apologies. Basically, what happens is you get these bands, and they look like an armadillo, or at least this is what Eric thought at the time. And you know, back in the day, fruit fly names were basically named after their phenotypes. So you know, there's some really cool, crazy genes that are named the Halloween genes, for example, or cheap date. That is the name of a gene, and if you mutate it in Drosophila, they become incredibly sensitive to ethanol, alcohol, and they get drunk, basically. That's why it's called cheap date. And basically, at the time, Eric just thought that this was the name that they wanted to give it. And so basically it stuck. And so did a lot of the original Drosophila names. But nowadays, we stick to the original gene names, which makes it a little bit more complex. So the actual pathway itself is remarkably complex, and so what I wanted to do was just to show somewhat of a simplified version to kind of demonstrate my point. So this is the cell, and basically cells are competent to respond and to receive these Wnt signals, and they do so via the plasma membrane receptors. So this is what we call frizzled, an LGR, and then there's this other receptor called LRP. The main gist of this part of the network is the fact that armadillo, or as I said, in humans and other mammals, the beta-catenin, in cells that aren't activated by Wnt, this protein is constitutively degraded. So it's broken down within the cells. And it's a good thing that it is, because it's so highly produced that if it's not broken down, then the pathway becomes activated too quickly. It has to be done in a very regulated manner. This represents the nucleus, and inside we have these terminal effector proteins. These are what we call transcription factors, which I'll describe in a little bit more detail on the next slide. And these are interacting with DNA, and these tell the cells what to do and what proteins to produce as a result of the Wnt protein. So in the activated form, the Wnt protein binds to both frizzled and LRP. We get the complex of all these proteins are sequestered, so they're brought up to the plasma membrane. And basically what this does is this liberates beta-catenin from its breaking down, from its degradation. It enters the nucleus, and then we get all this new kind of gene transcription, which then drives fate. Specification or stem cell self-renewal. There's a lot of different things it can do. And it's important to note that in most mammals, so if we're talking about humans, mice, primates, and all the other mammals that you can think of, there are generally 19 Wnt proteins, 10 frizzled receptors, and then there's 4 of these transcription factors. So there's a wide range of diversity, and one massively open question in the field is how cells can respond to such a diverse amount of signal. So in the 1980s, a very important discovery was made, because it's interesting to go from the outside of the cell to the inside of the cell, but then the signal has to go from the membrane to the nucleus. And it's an interesting question of how does it do this and how does it integrate this? So work done by Marion Waterman and Hans Clevers at the Utrecht Institute in Amsterdam, Holland, basically demonstrated that these TCF factors are important for this. And as I mentioned, transcription factors are these specialist proteins that interact with the DNA directly, and they regulate what genes need to be turned on and turned off. So, for example, if we take a stem cell and we want to make a muscle cell or a liver cell, then we turn on the genes that we want for the muscle cell, but we turn off the ones for the liver cell, and that's really, really important. Conversely, if we want to make a liver cell, we turn the liver cells on, but we turn the muscle cell genes off, and that's also just as important. If you don't do that, then the cells don't fate to the right lineage. They don't form the right kind of cells, and what you get are a bit of a mix, really, or you don't get the cell at all. The original TCF-left factors were actually found in immune cells, and this is why they're called T cell factor and lymphoid enhancing factor. They're originally from the blood cells, the immune cells. The long and short of it is basically is that they have this kind of wide range of evolutionary conserved domains, and then the armadillo beta-catenin protein, which I described in the previous slides, binds to this region of the protein, and its binding is absolutely important. These two figures here, these two diagrams here, demonstrate evolutionary conservation. So these are all different organisms. The C. elegans is a worm, D. melanogaster is the fruit fly, and this is Homo sapiens, which are humans. And what you can see, at least in this region here, is that there is a hell of a lot of conservation, even though that humans, or at least the ancestors of humans a long, long time ago, with common ancestor of worms and flies, they all share this evolutionary conservation. And again, another interesting question is how these have changed through evolutionary time from simpler organisms all the way up to humans, and how these are implicated in disease development, stem cell self-renewal. So it's interesting to understand how Wnt signaling proceeds, and a lot of work has then been done in understanding embryos. Because human embryos are quite difficult to somewhat obtain, they're only really obtained from IVF. Research on primates, especially in the greater primates such as chimpanzees and gorillas and orangutans, is completely banned. We, over the decades, have used the mouse embryo as a model to understand mammalian embryonic development. And what I would like to show here is that if we remove the Wnt3 protein from the mouse embryo, so this is a mouse embryo at around about 6.5 days of embryonic gestation, and what you can see here in the center is a lumen, and then this here is the cells that are going to go on to form the actual embryo itself. And the blue regions of each of these embryos shows the expression of the genes. This is a normal embryo and what it should look like, and this is what on the right is what happens when we remove the Wnt protein. So when we remove Wnt3 from these embryos, we lose this expression. T. brachyury is a gene that's very, very important for gastrulation. It's important for the development of the embryonic germ layers, so forming the guts, the lungs, the liver, and things like that. And if we remove the Wnt protein, at least Wnt3, we lose brachyury, and we lose other kinds of markers that are associated with Wnt signaling. So what we know from this is that Wnt signaling coordinates these events, and it's very, very important. If you remove parts of the Wnt pathway, if you remove its receptivity, its ability to communicate, it ends up in what we call embryonic lethality and actually ends gestation. And this is kind of a homologous event that would occur in humans. This would probably result in a miscarriage of what we call pregnancy cessation. However, to understand this in the human embryo, as I mentioned, is very, very difficult, not only to obtain embryos, but also because embryos in humans implant very far into the endometrium lining, and it's just not ethical to retrieve those embryos to understand this. So in terms of Wnt signaling and stem cell biology, we know that Wnt signaling is involved in the maintenance of mouse stem cells that I've shown you before. That was work that was done quite a while ago. As part of my PhD and my area of research, we're trying to understand what the roles of Wnt signaling is, at least this form of communication in human embryonic stem cells. But what we do know straight off the bat is that too much Wnt is bad for the cells, and they differentiate into these flattened cells and take on a different fate. So this is one aspect of my PhD, and the other aspect of my PhD is understanding how Wnt signaling affects early embryonic development in the human embryo. But also there's stem cells, like I mentioned before, in the adult, and these are all kind of very niche, and you find them in lots of different parts of the body. So the intestines is a really good example, the liver, the heart, the brain, the eye, the teeth. And these are very, very important for the regenerative capacity of the organs. So the intestines is highly regenerative. Cells only live for 4 days, but because the intestines is involved in digestion and absorption, it's a very harsh environment. So regeneration needs to be optimally maintained. And Wnt signaling actually is involved in this. So if we have a closer look at the example of the intestines, what we actually see in the gross anatomy of the intestines is that actually we have these, what we call villi, which are these protrusions that are involved in the absorption of nutrients. And if we have a closer look at the cellular structure of these cells, of these villi, at the very bottom here, where we demarcate the what we call the crypts of the villi, nestled in here are stem cells. And if we look even closer at these special cells, these mesenchymal cells, which are supportive, and what they're actually doing is secreting, releasing Wnt proteins into the environment, which maintain these stem cells here. And so basically, they allow for a controlled regeneration of the stem cell population and a controlled regeneration of the intestine, all the way through adult life and through the lifetime of any organism. And what we know is that actually, if we mutate or we remove the function of Wnt proteins in the intestines at this stage, at least in mice, mice die very, very quickly when they're born, because their intestines can't regenerate. And actually, if you look at histological sections of mouse intestines, you see that there are no villi at all. And because of this, we can generate these really, really cool things called organoids. And we do this by removing cells from a biopsy. We can do this from lots of different tissues. Actually, we can even do this from the venom glands of snakes now, believe it or not. And what we do is we take the normal tissue, we dissociate it, and then we place it into a special matrix with all the growth factors that are required. And what you get are these very highly organized structures that resemble the intestines. We can do it with liver, brains, you name it, we can do it. And this has been really instrumental for treating things like cystic fibrosis. And so what one can do is take these cells and grow the organoids from cystic fibrosis patients. You can take them directly from their intestines, grow them up in the lab, these organoids, and then you can treat them with a chemical called forskolin. And basically what this does is it causes the organoids to expand. So in this case here on the left, these are cystic fibrosis organoids from patients. And when you give them forskolin, they don't actually expand. But actually, if you go in and treat the organoids with a small molecule drug that's involved in treating cystic fibrosis, or if we use genome editing to repair the cystic fibrosis gene that's involved, we actually get this expansion. And so basically this kind of paves the way for something we call personalized medicine. So in the future, you might be able to go to your GP, give a biopsy, they go away, they send them to the lab grows up these organoids, and we basically test drugs to tell you which drugs are going to work and which ones aren't, which ones are the best for patients, because everyone's genetics are so different. So this is really, really cool, and this is where the field is kind of going. So to summarize this, we see that Wnt is really, really important for this. It's involved in health and disease and embryonic development. That the communication of Wnt is mediated by this beta-catenin protein, the armadillo protein that I showed you. But the most important thing is that it's under tight control at all times. We know that it's involved in human development. We know that it's involved in mammalian development, in mouse. I've shown you this before. And it's helping us to guide scientists towards treatments for infertility, cancer, and degenerative diseases. But whilst Wnt signaling maintains the longevity of these cells, these stem cells in the organs, what happens when this becomes dysregulated? So to go back to the original slide of showing you what the stem cells are, remember that these stem cells can remain undifferentiated, or they can differentiate. And then sometimes when stem cells become overproliferative, so they divide too much, One mechanism that the environment deals with this is by causing them to differentiate, because when they differentiate, their lifespan becomes locked in, whereas a stem cell can renew for the lifetime of an organism, or in the lab for indefinitely. So what actually happens is, and I kind of hope you might be able to piece this together yourselves, is that when stem cells become aberrant in their communication, they don't control it, developmental biology and cancer biology start to become two sides of the same coin. So this is a wheel of basically all of the different characteristics. I wouldn't pay too much attention to the outside, that's just the inhibitors, but the inside are hallmarks of cancers. And they can evade, growth suppressors, so they can continuously grow. They resist cell death. So when its DNA becomes damaged, one of the things that cells do is they undergo this thing called apoptosis, or cell programmed death. And that's really important because that removes these unwanted and potentially cancerous. They induce angiogenesis, which is basically where they start to develop blood vessels so they can feed cancer cells. But this all stems from overproliferation of the cells. So how does Wnt actually do this? So one really important thing and difference of Wnt communication is that during embryonic development, it's transient. So it comes on and then goes off very, very quickly, and it's very tightly regulated. And that's absolutely important because the speed of development is very, very quick. Cells need to be differentiated. They need to know where they're going, so it needs to be quick. However, that mechanism is absolutely not compatible with stem cells. Stem cells need a slightly elevated or an attenuated level of prolonged Wnt signaling to maintain their ability to self-renew. So I would like to think at this stage you might be able to see this implication that actually too much Wnt is bad for the cells, and this is where we start to actually get cancers forming. So whilst Wnt is really important for embryonic development, really important for maintaining stem cells, it's also really bad in the sense that it can form and lead to cancer. So it's important that we recognize that the Wnt communication that cells have between each other is controlled intrinsically by themselves. And in one case where this kind of goes completely skew with, it goes completely out of proportions, is in colorectal carcinomas, so cancer, of bowel cancer basically. And 80% of all bowel cancers have a mutation in this gene called APC. And APC, if you remember from the previous slides, but at least more simplified here, is involved in maintaining beta-catenin's degradation or its breakdown. And so what it's doing is it's lowering it, it's maintaining it, it's keeping it within a threshold. When you lose APC's function, beta-catenin becomes regulated and it becomes stabilized, and it goes to the nucleus and controls genes that it's not really supposed to do. And it starts to regulate genes that are involved in growth, evading cell death. And that's when you start to get the initiation of early stage cancers. It's hyperproliferation, hyperactivation of the pathway, something what we call hyperplasia, where we get lots and lots of cells that are dividing. And it's important to note that because APC in the cell is downstream of the plasma membrane, downstream of this level, the control of the cell, the control of its signaling pathway is completely lost entirely. But it's important to also note that cancer development is a continuum. It takes a long time generally for cancers to develop. At least in adults that we know of, cancers can take 2, 3, 5, up to 10 years to actually manifest in itself. Things like pancreatic adenocarcinomas, for example, when things like you start to get diabetes or other kind of insulin-related pathologies, this is from a later stage cancer when cancer is relatively untreatable. But here between the cancer, so the initiation in the intestine, between the normal lining of the intestine to where we get this hyperproliferation, here's APC2. And then as we move along through cancer, and then we actually get a colon carcinoma, where then it becomes metastatic. So when the cancer cells actually begin to metastasize to other organs, we see that these cells start to actually accumulate more and more mutations. So KRAS mutation, which is involved in FGF signaling, which is another form of cellular communication, P53, which is a protein that's involved in regulating the genome from apoptosis. you can see that over time, the cancer is a continuum, and actually it takes a long time for it to develop. But on the horizon, there are treatments. And from our studying of the Wnt pathway, we've been able to really start to understand how we can target each of these individual parts of the pathway. So as I mentioned before, Wnt proteins are released into the outside of the cell. They don't like it there. And the reason for this is this little tiny part of the Wnt protein is a fatty acid tail, and it doesn't like being in an environment where there's lots of water, basically. And so we can kind of block this, but the real problem with this method is that when we start to block Wnt signaling globally, it has off-target effects. So these porcupine inhibitors, known as porcin, whilst they block Wnt in cancers, they also block Wnt that's involved in bone homeostasis, for example. So people undergoing cancer treatments for Wnt-related cancers start to have problems with their gastrointestinal system, their bones, osteoporosis, and things like that. So we're trying to understand how these treatments can become more selective and more targeted. But on the note of the porcupine and how it regulates the exit of Wnt proteins from the cells, there is one notable condition called focal dermal hyperplasia, or GALT syndrome, which is caused by this disrupted Wnt communication. And the really, really quite interesting thing about this is the fact that the gene is located on the X chromosome. So females have 2 X chromosomes, males have 1 X chromosome and 1 Y chromosome. The Y, or the extra X chromosome, in the female comes from the father. He determines the sex of the baby, of the embryo, the fetus. And because this is X related, it's interesting because what we see now is actually that males, human males, with a germline mutation. So a germline mutation is basically passed down from through the embryo. It's not in the cells of the adult, are not found at all. And actually, this actually primarily affects females. So this is something really interesting, and it's definitely something worth having a look into a little bit more in the future and seeing if there's some more treatments, because a lot of skin conditions arise from this, and we get congenital abnormalities of the limbs, missing toes and oligodactyly and that sort of thing. So that's kind of really the end of the talk. And what I kind of hope to have demonstrated is that the Wnt pathway, this communication in cells and embryos and stem cells, really is kind of a Pandora's box. And the reason why I titled the talk a Pandora's box is the fact from the Greek mythology and things like this of Pandora's box is that it's a source of great but also unexpected troubles. But I think a much better interpretation of this is that it's a gift or a present which seems valuable but is in reality a curse. And I think for me personally, I do kind of view the Wnt pathway as that it's important for all of these different mechanisms, but it can also be a curse. So to kind of summarize everything that I've talked about today, the formulation of communication via Wnt signaling regulates both embryonic development and stem cells, but unchecked control leads to death of the embryo and degenerative diseases and cancers. The Wnt-driven cancers, there's lots of treatments for them, and we're making more and more better treatments every day, and the research is really pioneering for this. But there is naturally a massive and significant requirement to understand more in depth, which is what some of my research is doing and other people around the world. So just to acknowledge very quickly, we have to say thank you to Jenny for letting me join her lab and for all of her support and being in such a supportive environment from the lab itself. A big thank you to my course supervisor, Stefan Hobler from the University of Aberdeen. He's been really instrumental for helping me with working out what Wnt's doing. To the Cambridge Stem Cell Institute where I work, and thank you also to the MRC for funding this research. And again, thank you to the organisers for giving me the chance to give this talk. And that's it.
Great. Thank you so much, Connor, for an amazing talk. Just a quick reminder to those listening that if you do have any questions, please post them in the chat. And in fact, there seems to be one there already from Nick Alcock, who's asking about the embryonic cells that were in too much Wnt, that looked like they were almost epithelial.
Yeah.
Do we know what lineage they've been forced down yet? He suggests perhaps intestinal epithelium.
So yes, actually we do know really quite simply what these cells are. So when I was describing at the beginning of the talk the embryo, and you have the cells around the outside, the trophoblast cells, which go on to form the placenta, very, very important cells, of course. The inside, the little ball of cells that we generate the stem cells from, they contain two types of cells. So the epiblast progenitors, which go on to form the fetus, and then underlying as a little tiny band of cells called hypoblast cells, and they're really important for patterning the embryo, basically telling what's going to be its head and what's going to be its arse. And then they form the yolk sac. So actually in the human system more so, if we take the stem cells and we give them too much Wnt, or we kind of cause hyperactivation of the Wnt signaling, or a little bit more than what's necessarily controlled by the cells, we actually see that they adopt this hypoblast-like cell fate. So they don't quite form intestinal stem cells or any form of intestines. That would have to be done a lot later on in development of the stem cells to make those cells.
Interesting. I think I want to take it back a couple steps. So throughout the talk, you mentioned Wnt in a number of contexts, a number of times. I was wondering if you give a little bit more detail or maybe a bit more context in how Wnt changes within different cell types and why it's differentially regulated in that way.
Yeah, so the main premise of the Wnt signaling pathway as a form of ancient communication is indeed how individual cells can interpret the signals, basically. And there's many layers by which the cells can actually do this. So if we have the actual protein docking and binding with the receptor, the cell knows that this is happening, and so it will regulate the level of free receptors that the protein can bind to. The increase in the beta-catenin and the armadillo protein, and then its activation within the nucleus, is then also regulated by the cell. So there are many different levels of regulation. Another layer of regulation that these cells can do, which is also quite amazing, is that actually the cells can produce more proteins which bind to the Wnt proteins on the outside of the cell, and basically they bind in such a way that they block its binding to the receptor. And so regardless of the overall kind of amount of Wnt outside of the cell can effectively stop too much of the signals. And when you start to get dysfunctionality of specific proteins within the cell, is when you start to get too much activation of the pathway, and you get cancer, overproliferation, and cell fates that you don't want, really.
Yeah, makes sense. I think as well, one of the more interesting contexts you mentioned were organoids. And they're always incredibly interesting, as if you just look at them, they don't often look like the organ they represent, and yet they act so much like them. So you already touched on it a little about their application, particularly for targeted treatment. Mm-hmm. But what are your thoughts on organoids, particularly for the study of Wnt?
So like all models, that come from stem cells and things like that, they all have their limitations, and there's always something, that can be improved on. And this is the wonderful thing about science, is that it's constantly improving. And a model that was wrong, or not so optimal 20 years ago can be improved. Organoids are really fantastic in the fact that they can do a lot of really cool things. I think one very important thing that these organoids can recapitulate is the 3D environment to a certain extent. Cells generally act in a 3D way. They're constantly in contact with each other. One of the limitations of this is the fact that organoids don't have support from other cells. So we don't have immune cells, we don't have innervation, we don't have those sort of things. So there's a kind of a line where we have to stop. Organoids have been really instrumental for studying wind signaling, absolutely. We know that wind signaling is really important for generating organoids. It really makes no difference as to what tissue they come from, whether they are, cerebral organoids or organoids that come from the brain, organoids that are coming from the lung, the liver, the stomach, the intestines, the prostate, the uterine lining, the fallopian, you name it. As long as there's some cells that can regenerate and repurpose, their stem cells, we can take them out and we can derive organoids. And as I mentioned in the talk, Hans Klaver's lab, who discovered the TCF-left transcription factors, very recently showed that they can actually make organoids from snake venom glands. So they take the cells from these venom ducts within the snake, and Wnt is the main protein that's required for this. And then you basically make these organoids that produce venom. And this is amazing because we can use these organoids to study the toxins of venom, but also if there's any molecules that could have efficacy in treating diseases, but also for antivenoms for when people get bit by them. So the applications of organoids are really quite amazing. And for studying wind signaling, but not only just wind signaling, other pathways, they have been tremendous, really.
Great. I do not see any more questions in the chat, so I'll take this time to thank you again, Connor, for a great talk.
Yes.
We will be reconvening with our next talk at 1:30, which is How the Brain Learns to See, which will be done by Dr. Susanna Barret Mierau. And so I will see you all then.
Hello.
Hello, Susanna, can you hear me?
Hello and welcome back, everybody. We're going to be starting our next few talks in this afternoon of the Cambridge Festival. So our next talk will be by Dr. Susanna Barret. And I would like to remind everybody again that if you do have questions, please include them in the chat. I'll be reading it and can ask your questions at the end of the talk. And then, so without further ado, I'll pass it on to Susanna.
Good afternoon. My talk is titled How Babies Learn to See, and there may be slight lag between some of the videos and when they're played because it's going over the internet. So I'll try to take a pause there. Also, some of the videos are a little bit small, so if you have the option to make your screen, the video full screen, you'll be able to see better. So this lecture will cover how vision develops over the first year. Here we have one of the remarkable things here is our son when he was just a few days old, and our daughter is day 2. So you see that it's not just vision, but the actual size of the brain is increasing quite dramatically over this period. And we'll especially focus on the areas of the brain that process information that comes from the eyes. So many people may be aware that we are born basically what we would call legally blind. This means our visual acuity, or how far we can see away things in focus, is about 3 over 60, which would be 3 meters, seen at 3 meters, what others could see at 60, or in the United States, seen at 20 feet, what others could see at 200. But and this is about the distance from the breast to the mother's face. So the baby can see the mommy's face in clear, but not across the room. And this improves over the first few months of life, until we achieve the adult level of acuity. One of the other features about learning to see in early life that's really critical to the later stages is the ability to move the 2 eyes together. So here we have our daughter at 1 week old. And if you look closely, her right eye here, you can see the pupil is looking straight ahead, where the left eye is looking over to the right. But you can see that, and if you've ever watched a baby in the first few weeks, you'll see sometimes their eyes move together and sometimes their eyes move independently, and they move more and more together over the course of the neonatal period. And here you can already see at 2 weeks, here's an example of our daughter looking the same direction with both eyes. And this is really being able to coordinate the eye movement is really critical for developing a visual skill, which is called depth perception. Now, some of these old time 3D pictures are those who were, children in the 70s and 80s may recognize this Viewmaster, but the idea is because each of our eyes, if you open and close one eye, sees a slightly different view, the brain learns to fuse these images over time. And so that's why by separating the 2 and showing these pictures, you get that 3D perception. Now, scientists have studied the development of this depth perception in babies. And one of the most famous experiments was the visual cliff, here shown from the 1960s. And here the experimental apparatus is there's basically a platform with the strong check pattern, and then it actually extends as clear plexiglass. But the baby can't see the plexiglass, so then the mother stands on the other end and calls to the baby. And if the baby doesn't perceive a big drop, the baby will happily crawl across to the mother. But if it perceives a big drop, it may be hesitant to do so. So just a little warning here, do not try this at home. However, there are ways that you can look at the depth perception developing in a young infant of your acquaintance. So here you can see our son at 6 months. You can see he's already looking, kind of reaching out and trying to calibrate this depth perception. Here he wants to get a little spot of tea. And here I have a little video. So what I want you to see here is the sensory system develops first, and then the motor is kind of relies on that sensory. So you can see, even though he's not the fastest at grabbing the food, he did grab towards the back of the tray versus the front of the tray, showing that he has developed some of that depth perception. Here you can see another one. Here I have the, little puffin. You can see him reach several times, and it's not perfect yet, right? He's still developing this integration of his visual information from his eyes and the movement of his arms, but he is able to estimate that depth fairly well. As children get older, they learn to judge even larger distances. And this leads in part to this famous separation anxiety, where the baby starts to realize that mom can get away or dad can get away, and starts to be quite upset when the parent's away. But it also leads to exciting things like cruising. So here, our son, he's not able to walk on his own yet here, but by trying to judge that distance between the coffee table and the sofa, he's starting to be able to use that depth perception to aid in another motor skill, which is walking. So it turns out the early child period is a critical period for learning this depth perception. If one eye is able to move, for example, a problem with the muscle, or one eye doesn't see as well, for example, if they're a cataract or some other problem with visual acuity, then the brain cannot learn to fuse these images together. Fortunately, in the early child period, if this is recognized early on, you can actually patch one eye. In fact, you patch the good eye, which forces the brain to focus more on the weaker eye. And this allows children to, obviously we can take the patch off, to start using both images from both eyes to develop this depth perception. So how does what the eye sees reach the brain? So if you had seen me give the public lectures for the talks in the year before, this is the picture of the PDN lecture theater. There I am down at the bottom. And if you were looking at me right now, or if you're looking at me on the video, the information is coming into the eye, and the part that really helps us know what we're seeing is located, ironically, in the back of the brain. So here's the eyeball in the front. So this is like the forehead, and this is the back of your head. And actually what happens is, if you look here, when we're looking at a visual scene, the information that comes from the right part of our world, we call that right visual field, which here is depicted in green, hits the nose side of the inside of our eyeball on the right side of our head, and the outside on the left. And that information is carried to the left side. So the left side of the brain pays attention to the right side of the world and processes information, and the information from the left side of the world is processed on the right side of the brain. And so we're going to really focus on, so when this information comes from the eyes to the back of the brain, what is it doing? And unfortunately, we know a lot about this location because of injuries. In World War II, the British helmet design did a good job of protecting the top of the head, but unfortunately left this part of the brain in danger of trauma. And so here you can see in 1918, this report where they documented which parts of this area of the brain were damaged and which parts of the visual world could no longer be seen. So one of the biggest advances in really understanding how the brain processes information from the eyes came from two scientists, Hubel and Wiesel, who really discovered that the brain it reconstructs the visual world. And what we're saying is we have this perception that, it's kind of like a movie projector coming back, that what we see when we look out in our eyes is an exact reflection of the world around us. But it turns out that this is not true. What we see is what our brain has reconstructed from the information in the outside world. And in their experiments that they started in the late 50s through the early 1970s, for which they received the Nobel Prize in 1981, they determined that when they were recording from brain cells in this back area of the brain and showing the cat different pictures, the brain cells responded not to the different shapes or things they tried, but actually when they were pulling the slides out, the orientation of the line. So some cells might prefer a straight edge versus, this many degrees to the right or the left. And what they learned, and what's very exciting, is that the brain starts to learn by detecting edges. And so when the information comes from the eye to the back of the brain, you're basically getting very basic features, breaking down to the world into its most basic components, such as edges, orientation, contrast, color. And so what the baby's actually seeing in the first few weeks, if you see the little mobile we got, is probably something more like that. It's still interesting. There's edges and contrast and color and all these, but the baby hasn't really started to put it together into what you and I see. One of the lessons I learned from our child is that we got this cute little baby lounger to put in the living room. And initially we oriented it like the chairs and sofas, so it faced the middle of the room. But our daughter had very little interest in the first few weeks in sitting in it. But we noticed when we turned it around so that she faced the curtains, she was very happy and she'd sit there for ages. And of course, the curtains that happened to be in the flat we lived in at that time in Cambridge had this very striking high contrast pattern. And once we realized this, that I applied the visual neuroscience I knew, it was very clear why this was interesting to her. The brain also learns to then take these edges and basic components and then see motion. And it's that motion that helps us determine what belongs to what that allows us to see form. And so here, these edges and contrasts lead us to form corners and shapes. And this, we also had the funny thing in the first few weeks where we also had this little swing. And we had this fantastic little toy here to attract the baby's attention, but we found that she was often looking off to the side. And then we realized that she was looking at the arms here because when the swing swung back and forth, that stayed steady, but the other stayed the same. And so that motion was really attractive to her in the first few weeks. And either that or she had an early interest in mechanical engineering. So once the babies can perceive form, then objects can catch the baby's interest. So here you see at 2.5 months, she's discovered she had an arm. And if you watch babies in those first few months when they discover their arms and legs, it can entertain them for hours. The arm is a toy that will hopefully not fall off. Here at 3 months, you can see she's finally noticed all these toys that are there. And she's able to distinguish that there's something interesting there. She's maybe a little bit confused why the elephant is turquoise. No, just kidding. And by 3.5 months, she can perceive other shapes, such as reading a book. Okay, that's a joke. Okay, the brain attaches meaning then to objects. So it's not necessarily by 19 weeks, but just to give you an idea that as the child is progressing into months, they start to be able to identify different objects, especially objects that are really important to them, like their parents, favorite items, things like that. And so here, as the areas of the brain that surround so if the basic information is processed here, it's the areas that surround these associated areas which start to put these together so that we get corners and shapes, and then these can eventually be combined to have meaning. Now I've put the words here for us, but obviously it's even later that you get language being incorporated in with the visual. So here's something you can try at home. For recognizing objects. So here is our son sitting at the table. And I put these little food items, which are freeze-dried little vegetable things. And you can see he goes right away, even though there's, the tray is there, he recognizes right away that there's something different than just the tray. And even though his motor skills are not up to being able to necessarily grab them very easily, he's able to see that there's something different there. So he recognizes that there's an object. So one of the things you can do to look at this is can you alter the contrast? So here there's a good contrast between the food and the tray, but now let's try the next part. So here in the top left, we have a low contrast, right? Where you can see that there is a little piece of food there. And when I play it, he's looking around, but he doesn't see anything interesting, and then he gets distracted. And so you can see. Now you can see, if you look before, now if you increase the contrast, so now it's on the purple tray and here, then you see he grabs it right away. So he obviously sees that there's an object there. And even though he's not that interested in it, he still grabs it right away because he can recognize that there's something there. On the other hand, you can also try difference between preferred and clearly non-preferred objects. So here he's looking bored. This is not his preferred object, but look what happens when a piece of croissant from Maison Clément gets in there. This is a high preferred object for other people in our family as well. And you can see he immediately recognizes the difference between these two objects, right? They're both something different than the table, but this is very exciting. And then you can see another example here where, you know, he immediately identifies not just that there's an object, but something that he's interested in. So clearly he's already attached some type of meaning and recognition of these objects. You can also try this at home by looking at the favorite books at different ages. So here's our daughter at 7 months, and here the books that were most attractive to her had a simple white background and a single object with high contrast. And especially for her, up until this time, those books were the most interesting to her. And as she was able to understand more about the meaning behind the different objects and to understand contrast, we have books with more simple, they're still fairly simple, but now they have little situations, and you can see she finds this highly interesting. And then a book that she had very little interest in at these younger ages, as she became older, it has a more stylized picture here, and she became more and more interested in it as her knowledge of the kind of complex visual social world came into place. And then one thing that was super interesting to me was I had got this book about animals in the prairie, and it's very artistic. You know, one of these where all the pictures are put together with little pieces of paper. And she showed no interest in it prior to a trip when she was about 1.5 years old that we took to Yellowstone National Park in the American West. And then when we came back, she just picked up this book and would look at it all the time and identify with, you know, the bison and other animals that we had seen on the trip. So that was very remarkable to me because it was clear that she had learned things on the trip that were now influencing her interest and recognition. But it's not simple enough for the brain to just see and attach meaning, but we actually have to know where to look. Some of you may recognize these kind of busy scenes where then you're tasked with finding something in there. If I asked you to find a sailboat, you probably almost within seconds immediately be able to identify where the boats are, because, you know, without either consciously or more likely unconsciously, you already have perhaps interpreted this as a beach scene, equating the yellow with the beach, the blue as the water, and the lighter blue and white as the sky. And so if I say boat, you immediately look for the boat in the water. And if I say, can you find the horses? You know, you might have to look a little bit more deeply, and you might even take kind of a scanning approach to find them. They're right here in case you're wondering. You can try this at home. So here's our daughter when she was 2, and she had this bookshelf here. So I noticed when I would say, oh, can you find, this book? If she remembered the exact location, which she often did because children at this age can have exceptional memory, especially for spatial locations, she would be able to find it. But if I asked her for something non-preferred or had been moved to another area, it often took her a long time to find it. If I said to you, oh, can you find Paddington Bear, or her stuffed Paddington Bear, you would likely look immediately at the shelf right here because you've already perceived that these are books and these are stuffed animals, so you're more likely to find the stuffed animals. And so as the children develop, they can use this memory to start to make predictions, and then this prediction can help them target that visual search. You can look at your own child's visual search by using any book. Here's a popular scene from a popular book, Goodnight Moon, you know, where you could say, find the kittens, right? And then just watch the child look. You know, do they start looking over at the top of the scene? Do they look out the window? Do they look on the floor? And you can make it more challenging by, asking for smaller details. One of the things too is because if I ask you to scan for small details, you might take a more ordered approach, or you've developed strategies for you either to predict or to systematically look for things. Because children are still developing these things, it takes them longer. And I'm often surprised, especially with a toddler, where I think they hadn't paid attention to the question. And then if I actually just wait a little bit longer, then they point to where it is. And I realized that they were trying, it's just taking them longer. And part of that is due to this control by the front part of the brain that is still developing. You can also see this with older children in counting objects. Initially, when my daughter was learning to count, she knew her numbers and understood the concept of counting, but she didn't have that ordered. And so she might take them out of sequence, which could lead to either double counting or missing one. But as she's become older and developed strategies to count more, she might take what you or I learned to do, which is a convenient convention of, in this case, reading left to right, and there's a higher probability of counting without missing or double counting. Another aspect of brain function that's for vision that's very important is that the brain only sees what it's looking at. I think conceptually it's quite easy for us to understand that we can improve our ability to find things visually if we order ourselves and use the kind of pilot in the front of the brain to guide. But I think very little of us are aware of how actually we are only perceiving what's in our center vision, and we actually have to look around. We have this perception that we see the whole scene in front of us, but it's actually many magic tricks rely on the fact that we believe we see everything that's happening when we're actually only processing to a great degree what's right in front of us. So in this famous picture, all the way back from the 15th or 16th century, you can see here, he's got his trick going here, and everybody's paying attention. And then this little child is, likely picking something that perhaps will then be used in a later trick. And because people don't think, oh, well, anybody's been near to me, it seems like magic, but this is actually taking advantage of this disconnect between what we perceive and what's actually happening. And the way the brain constructs the world when it can only pay attention to small parts is to move the eyes really rapidly. And these little jumping movements or saccades are so tiny and so fast that we're completely unaware of them. But if we're looking, for example, at this famous photo here, this is the lines drawn by a computer software that basically tracks the eye movement. And you can see that most of the time, the eyes are looking at the eyes and also at the facial expression. That's where we get a lot of information as humans. There's this cursory look around the edges, but you can see actually little to no attention is paid to this area at all by the eyes. And so here's a scene from Yellowstone with all of the bison resting on the open plains. And, we have the perception that we see everything really equally here, but actually the brain is using these tiny brief eye movements to take little snapshots and reconstruct the visual scene. Now, some of you may think, well, I'm into photography and this kind of stitching, where we take multiple photos and then stitch them together. That makes sense. That's what the brain's doing. But actually, that's not what the brain's doing, because the brain is actually doing something like this, where it's spending a lot of time looking at whatever, is decided to be the most interesting, and spending almost no time looking at the rest. Yet this is what we perceive. And so you and I, looking at the same picture, might actually see something quite different, because our brain is actually maybe choosing different aspects to focus on. So here, this is a little harder to try at home, and there could be multiple interpretations for this, but here you can see our son is quite interested in some object in the kitchen. And you can see that when my hand comes in, he says no, and he pulls it away. And so here he's still got it. But when another, here, so you can see, yeah, so here you see he has the toy, he sees the hand coming in and he's like, nope, he's pulling away. But look what happens when another object catches his attention. Here he's still holding on to the other object, so we assume he wants it, but he doesn't seem to even notice that the other hand has come in. Now you could say alternate interpretation is he didn't want it anymore, that could be it, but he really doesn't seem to see the hand that's kind of reaching in for the egg at that point. So to sum up what the talk has covered so far is you can see that what babies see over early life really changes dramatically in the first few years, where at birth they don't see very well, they have to work on moving the eyes together. And once they can see in focus, detecting those edges, that allows them to detect simple patterns and then use those patterns and the transitions with the movement to interpret form, then to recognize objects. And then once we have objects, to start to understand categories such as animals and group objects by meaning. And then by being able to understand meaning, attach meaning to these objects, we can start to notice patterns that develop in the world or sequence of events so that we can predict things. And we can start to guide what we look at to help us navigate our environment. So just to sum up, that what babies learn to see is both interpreted and directed by the brain. It involves a lot of brain areas. And I just wanted to say thank you to the Cambridge Science Festival for hosting the talk, to the organizers and volunteers in our department at the Physiology, Development, and Neuroscience. Thank my children for teaching me and a lot about visual development, and also had the pleasure to know David Hubel when he was a professor emeritus and I was a medical student. And he's really a pioneer in this field and very inspiring.
Thank you for an amazing talk. I cannot wait for your kids to grow up and see this presentation. They're going to love it so much. So just as a quick reminder, if you do have questions, please feel free to use the chat and I can see it and ask the questions. I'll start it off though. One of the interesting things that you showed was the eye tracking, and in particular how we look at a face, for example, and what we focus on. Has that study been done with babies, and are the patterns similar or change as they grow up?
Yeah, it's actually very interesting. They have been done with babies, and one of the advantages of eye tracking is it doesn't require language and it doesn't require people to follow. Well, I mean, ideally the head is oriented towards the camera that's doing the tracking, so I guess there is some. But they have done it, and it's actually quite interesting. One of the studies that was done is they show like a little video, to a baby, and then they look tracking over time. And actually, how much time the children spend looking at the whole environment versus the eyes does change over the first few months. And I think that's also indicative of how we start to learn and make predictions, changes the interest and the percentage of time that we spend looking at different either parts of the face or other pictures.
I guess kind of along kind of a similar line of how babies initially start to understand their environments. One thing that caught my eye from the cricket example when counting is that the crickets are always at different perspective. So is there a point where babies start to understand that looking at different angles is actually the same object?
Yes, I think first of all, key is to be able to identify form. That's something, say my hand here is distinct from the other parts of my body, or the virtual, well, I guess it disappears into the virtual background there. So once you start to distinguish form, because the motion is very powerful, then I think babies do start to recognize different parts. And there are actually cells in the brain that are kind of located in these areas between, say, the parts of the brain that that process information from the eyes, and parts that process sound, and particularly language, that will actually respond to pictures of people from different angles and things. So there are these integrative areas of the brain that help us say, okay, that's mom in side view or mom straight on.
Yeah, and I think as well, when your son was grabbing onto the contrasting object and obviously had no interest in it. That instinctual grab was very interesting. So it made me start thinking, how much of that training to see contrast is independent of conscious thought?
Yeah, so I had a professor when I was an undergraduate at MIT who in the last lecture for the introductory, neuroscience course is basically said, we have this perception that we're one person, but we're actually, many people that, have different desires. And I was like, whoa, this is getting very philosophical. But then I sort of understood what he meant, because we all have conflicting desires, right? Like right now you may be part of you is enjoying watching the YouTube, and part of you is, needs to go to the toilet or, things like that. And so we're constantly having to balance these needs. And I think we see this in little children, right? Because they don't have the same emotional regulation as adults, right? Where sometimes there can be very competing needs. And so, like grabbing things around him is a great desire at that age, but also he's like, really, he doesn't really like that particular snack. And so I think there's those kind of mix, or the egg timer. The egg timer is super fascinating, but nothing tops the iPhone apparently.
I think that's something we all agree on. I think there's a question in the chat from Sounds of Silence. It seems that the conceptual understanding started to come into play later in infancy. How do you study this kind of thing? I.e., how can you know if conceptual knowledge is in play?
Yeah, so the field of developmental psychology has many fascinating kind of experimental paradigms to be able to try to get at these concepts. I think that for me, it was quite fascinating to really look to see how these play out at home. When I had my N=2 longitudinal study where I got many data points throughout the day. And I think here, I think this recognition of categories was one of the earliest things where I was really confident that they had some, kind of concept or contextual information. The other thing too is the development of language for children who speak early can also be quite. One of the most fascinating things to me is because we lived in Cambridge and my parents lived in America, is that I often wondered, is speaking on video chat or FaceTime with my family, what do my children have a recognition of that? But when my daughter was pre-verbal, my father would always make this kind of clicking sound. And then she could when she started to be able to make that sound, which she could before she could speak, when she saw him, she would make that sound. And so I think she clearly not just recognized him, I think that was evidence of that, but it associated him with a different sensory modality. And this evidence of this kind of sensory this sensory social integration coming within the first few months. There's some exciting experiments done where they show either videos or play sounds that are either something relatively non-social like water running versus somebody coughing or sneezing, and already they can see that babies respond differently to the sounds that are associated with humans, kind of social sounds than ones that are kind of neutral, neutral social content. So I think there's several different ways that we can find this. I was also very shocked with our daughter before she was verbal too. Once I said grandpa when I was talking to my husband, and she made the clicking sound. So clearly she had, associated, the seeing somebody making a clicking sound repeatedly on the FaceTime with his name, grandpa, and then understanding that we could refer to him in different conversations, So I think the other thing that's surprising with kids is that because we generally judge people's cognition incorrectly, right, by their ability to articulate verbally or by their, physical responses. And of course, speaking is a physical response, right? It's like one of the most fine and difficult motor tasks that we do is talking. because you have to coordinate the lips, the tongue, the soft palate, the larynx, the lungs, in all very fine and rapid succession. And so because we often judge perception, like what the person's understanding and perceiving of their internal perceptions of the outside world by what they can express outwardly, I think we often really underestimate what children are doing.
Yeah, that's amazing to hear, that association from something that's not even a real person per se to the verbal.
Oh yeah, and I think I'm sure now with the pandemic, many other people are experiencing this as well. But my children, like we only went a few times a year to America, and I definitely felt that they developed a relationship with their relatives, in America, or with my husband's relatives in Sweden, over that really built, and they recognized them in person and made an association between what they had seen on the call and the person that they saw in person. And so I thought that was also really remarkable because, even more complicated than side profile or front profile, but to understand that, somebody can be on a screen and in person is apparently more intuitive than to some of us who are older.
Yeah. We do have another question from Hugo Smith. Thanks for a great talk. To what extent are the functional divisions of visual cortex defined before birth, or do they form as the baby becomes more experienced at visual processing?
So I think this is a great question, because this is really where Hubel and Wiesel, who started recording from the cat brain, wanted to know what are these cells doing, and what are they responding to, and how does the sensory environment influence that development? And prior to their work in the 1950s, the brain as a whole was kind of seen as this blank slate they call tabula rasa, right? This is just like written on by the world. But actually what was found is that there are these different brain areas and cells that are primed, waiting for specific types of information. And then they use, for example, the early visual information to really tune their cells. So initially a cell might respond to many different visual aspects, but over time it shows a strong preference. And those strong preferences for different types of input allow for, different types of complex functions to form. And what's really fascinating now is that because of the paradigms that were used in the second half of the 20th century to understand, how the brain processes visual information this way, now as people vary the types of stimulation they give, we can see even more and more different varieties of function that these types of cells can do. So now, making predictions about movement and all sorts of things, receiving connections from other areas of the brain that help shape this, our kind of understanding of how we process and understand the world around us keeps expanding. And I think that's one of the really exciting parts of neuroscience to me, is that there's just so much more to learn and also to understand at different levels, right? This whole talk talked about how to, looking at the behavioral level, what are babies doing? How can we take a guess at how they perceive the world based on, their behavior, watching their behavior. But, we can also, people use what's called neuroimaging, right? Using a magnetic resonance imaging scanner or other technologies to try to look at brain area activation. And then using either animal or cellular models, we can also see, how does this happen on the individual level of the brain cells or groups of brain cells, which act in small networks. So I think and then of course there's molecular and genetic levels as well. And so just kind of across scale as well as across the lifetime, I think there's just so much more exciting things to learn.
Absolutely. I don't see any more questions. But that was an amazing talk and great discussion afterwards. So thank you again for coming and speaking. And we will be reconvening in about 15-20 minutes with our final talk, which is Lessons from Labradors on how genes cause obesity. And I will see you all then.
Great. Thank you very much.
Hello and welcome back, everybody, to our last talk of today's session. So we will be hearing very shortly from Dr. Eleanor Raffan, who will be telling us about the lessons from Labradors on how genes cause obesity. So I will pass it off then to Eleanor.
Thanks, Dylan. So just to check, can everyone see you can see my screen, and we're good to go. Lovely. Well, it feels so funny. Last time I did one of these science festival talks, we were in a proper big hall, and it's such a shame not to be able to have that same atmosphere today. But hopefully this format means that more people can reach this and see it over time. So it's very nice of you very nice of those of you who've come along live, and hopefully a few more of you will watch later on. My name's Eleanor Raffan. I am a vet and lecturer at the Department of Physiology, Development and Neuroscience. My background was very much as a clinical vet, and then I kind of got the bug for research and science, and I've been very lucky for the last few years to be able to combine those two things in studying dogs, a little bit like Woody here. I love this little video that came from a friend of mine. It was her dog, and over his life, Woody got into all sorts of scrapes in the pursuit of food. I don't know how the timing on the videos is. A previous speaker said they were a little bit off, but you probably got that cute little whimper at the end. And he was an absolutely typical Labrador, really highly food motivated, always on the lookout for a scrap of food that would satisfy his food craving. And I think studying dogs like Woody is well worthwhile, because as a vet, I can see the clinical importance of weight gain and obesity in dogs. But primarily, I'm also interested in contributing to the study of human obesity and our understanding of it. And that's really important because there is a global obesity epidemic, and obesity gets a lot of attention in the news, and rightly so, because when an individual gains weight, it puts them at much higher risk of a whole bunch of different disease conditions, including coronary heart disease and stroke and liver problems and high blood pressure. And all of those conspire to cause shortened lifespans and real impacts on people's quality of life as they go through life if they are overweight. And so certainly my funders are more interested in understanding human obesity than pet obesity, but why would anyone support really the study of dogs? And that comes down to the fact that actually in biology, we use what we tend to call model organisms a lot. The most common model organisms in which we study biology relevant to human disease are lab mice and rats. Even fish, fruit flies, and little worms are very valuable to us understanding the fundamentals of biology and very clinically relevant things as well. And what we can do in animal models is study conditions systematically in a way that we simply wouldn't be able to in the human population. Now, compared to these lab animals, studying dogs as a model organism is somewhat less convenient, quite frankly. I have no interest in studying animals in a sort of research. I don't want to keep dogs only for research, and so all my work is done with people's pet dogs or working dogs. Those kind of free-living dogs, although it's much harder to control than the lab animals that you saw on the previous slide, actually have huge advantages when it comes to studying human disease. And that comes down to two main categories. Firstly, the similarities between dog and human diseases. And secondly, their profound differences when it comes to dog genetics. So first, those similarities. Our pets, our dogs in particular, develop many of the same diseases as their human owners, and they do it while sharing an environment with them. That means they're exposed to similar toxins, environmental factors, but also to many of the kind of more high-level environmental factors, like the increasing availability of food in our modern world. And certainly, my patients develop many of the same diseases as humans, whether that's asthma or hip arthritis that needs to be treated with a hip replacement. And sometimes they do it in a way that is particularly convenient and informative when you want to study disease not only for the dog's sake, but also for humans. So for instance, this X-ray on the bottom left of the screen shows a kind of bone cancer called osteosarcoma, which is a pretty devastating disease in dogs, and unfortunately is really quite common in large and giant breeds of dog. In contrast, in humans, it's a very uncommon kind of cancer. And so there is real potential in studying something that happens commonly in our species, in order to understand a bit more about its biology relevant to humans too. And the same goes for particular kinds of heart disease, which unfortunately are very common in some dog breeds. And those similarities are certainly true when it comes to obesity. This kind of cuddly pair, you might generously call them, two Labradors who are both really quite overweight. And unfortunately, they may not flag up as looking that unusual to you. We as a society have got quite used to seeing overweight dogs, and the surveys that have been done most recently suggest that as many as 2/3 of pet dogs are overweight or extremely overweight, which we tend to call obese. And that's a problem for dogs because it causes disease and shortens lifespan. That's the basic take-home message. Now, it's pretty intuitive, really, that a dog who looks like the one on the left isn't going to have as much fun and have as high quality of life as a slim, athletic beast like the one on the right-hand side. And some of that is about the ability to be athletic and express natural behaviors. But a lot of it is also about the fact that overweight dogs tend to have a higher rate of quite a lot of different diseases, either with obesity causing or exacerbating breathing and heart problems, trouble with arthritis and other orthopedic conditions. Some hormonal problems are more common in overweight dogs, and some very practical things like incontinence is worse in overweight dogs. And that's something that's quite common in my middle-aged female patients. And if they become overweight, then it can really make living with those pets really problematic. And so I study canine obesity because of its importance in my species of interest. But I study genetics in particular because of its relevance and what it can teach us about human biology. And whenever I give a talk about the genetics of obesity, it's kind of important to say up front that, well, we know the answer to this question, don't we? We know what causes obesity. If any individual, human, animal, eats more than they burn off during their daily lives, if they have energy intake that is in excess of their energy output, they will ultimately pile on the pounds. And that's basic physics. It's no surprises. There's no surprise in it, and it's something we've known for a long time. But what's interesting is when many of us have the ability to overeat, why do only some of us gain weight? And what we know is that a lot of that variability is down to our genes. And this was initially most intensively studied in human populations, whereby studying twins and families, and more recently, very big populations, we can get very accurate understanding that the tendency of each of us as individuals to gain weight and to become overweight is heavily influenced by genes. 40 to 70% of that heritability of obesity has a heritable nature. And actually, that number is remarkably consistent with about half or more of an individual's tendency to gain weight being down to genetics, whether you look at broiler chickens growing for meat, or whether you look at cattle or pigs, or as it turns out, when I look at dogs. And so genes are important, but which genes and why? And why on earth would we have a genetic makeup that predisposes us to a debilitating problem like obesity? And the answer to that lies broadly in the fact that for the majority of evolution, dog or human, we have existed in what I often hear called a resource-poor environment. You know, the dogs in this picture, the humans in this picture from a cave painting 12,000 years ago really had very little worry about becoming too fat, but really did have to make a big effort to get food by hunting, by consistently gathering small morsels throughout the day. And certainly, if we forget to eat, then we die. You know, there's no question about it. And so, consequently, the body is well set up to remind us to eat, and actually to remind us to get high value food, high energy food, with a physiological setup that tends to promote us being a bit heavier than would classically can be considered to be healthy. Because remember, in the past, periods of high food availability were commonly followed by periods of fasting. And in that context, it makes sense to have a little bit of extra energy laid down as fat to draw upon when food is scarce. And so the system that I've drawn here on a dog diagram actually could very easily be, in fact was, adapted from a human similar diagram. The systems are highly comparable between species. And what we now understand after many years of intensively studying these kind of systems in research environments is that the body monitors the energy reserves that we have in fat by sending kind of long-term signals of the body's energy storage depots in hormones, specifically leptin, but others too. And then also sends out short-term signals from the gut and the pancreas, from the digestive tract, in both nerves and hormones, which give an indication of the energy flux from minute to minute, hour to hour, day to day. And those short and long-term signals are integrated by an area of the brain called the hypothalamus, which coordinates them, and kind of the net effect of those signals is translated into the kind of feeding desire to feed, or desire to stop feeding, or the sensation of satiety that motivate our feeding behavior, and to an extent our energy expenditure as well. And of course, all of that occurs subject to environmental influences about what food is available, what it's likely to taste like, how rewarding it's likely to be. And here's the bit that I'm interested in. Every level of that biology has the potential to be tweaked by genetic mediators, which can alter one way or the other each aspect of the system. Now, as for which genes are responsible and why, well, you know, there's been a lot of effort on that over the last 20 or 25 years. There's been much insight from the study of human patients and laboratory animals who have severe early onset obesity caused by a single gene mutation. And in those cases, obesity often starts early in life and is quite severe. And studying those patients has been very informative and very important clinically to improve their health outcomes. But the take-home message from that vast body of study out there is that kind of common obesity, the obesity that is tends to kind of creep on us as we enter into adulthood and then stick with us and get a little worse through middle age. Common obesity is broadly under the control of hundreds, possibly thousands of genetic variants across our vast genome. And where we have been able to identify which genes are responsible, they mostly affect how the brain regulates our eating behavior. and they are expressed, the proteins that the genes encode are expressed in the brain and probably modulate our perception and response to food and drive to eat. But there's still an awful lot we don't know. The largest what we call genome-wide association study done to date in about 700,000 people was vast and extremely powerfully set up statistically to discover new genes. It's found about 700 different regions across the genome. But even so, we've only explained less than 10% of the heritability of obesity. So I entered this field knowing that there was this question about which other genes are important and how they exert their effect, and proposed to answer that question by studying dogs. So you might ask why, very sensibly. I've told you that dog obesity is quite similar, but actually when it comes to genetics, it's the differences that are most important. I started out with Labradors. This is a single dog breed, a very common popular breed in the UK, and the nation's fattest dog breed as well. These guys come with a phenomenal reputation for being kind of real chow hands, very foodie dogs who will always be on the lookout for food, like that video of Woody I showed you at the beginning. And the reason I was interested in studying them is because the way that dogs have been bred since domestication from the wolf somewhere around 30,000 years ago has led to real benefits for geneticists interested in finding disease genes. So following domestication, there was a long period where dogs were largely free breeding. Yes, they lived alongside humans, and yes, humans may have encouraged them to produce different types with function in mind. So little short-legged dogs to go down rabbit holes, or big, strong, brave dogs to fight off wolves. But although they may have formed what we would recognize as breeds, these dogs were ultimately bred for health and with a job in mind. And so if there were health problems in them, they tended to be fairly quickly eliminated from the breeding population, or those negative traits not accentuated. In Victorian times, dog fancying became a fashion. And those breeds of dogs, which had developed over many thousands of years, were recognized, lauded, praised, shown, and importantly given prizes. And people desired a degree of standardization, because in that competition process, in dog showing, people wanted to know that their dog had the right characteristics, or that they could be breeding dogs with the right characteristics to bring home the rosette or the trophy from the next dog show. And in the process of formalizing what were called breed standards and formalizing dog breeding in recording it in a stud book, the Victorians unwittingly caused what we refer to as geneticists as population bottlenecks. And so that really rather a few founder individuals were forced through a narrow funnel into and became the founder populations of modern dog breeds. And that's had some really quite important consequences for the future. So we now have these extraordinary and really quite extreme diversity of dogs with different sizes and shapes, different personalities, different fur types, all of which are very similar within one breed, but very different and diverse across the breed. And from a geneticist point of view, the existence of old bottlenecks many thousands of years ago at the domestication from the wolf, and these recent bottlenecks at the point of breed formation in the Victorian era, has led both to problematic ill health, which isn't something I've got time to dwell on particularly in this talk, although it is important to say that the high food motivation and obesity prone nature of Labradors is part and parcel of that. not only do we see those ill health things, but actually it's got somewhat of a flip side of that, is that there are benefits, because as geneticists, we are able to track down disease-causing genes rather more straightforwardly in dogs than you can in a very diverse human population. And that comes down to the fact that when we go looking for the gene or the mutation causing a disease, essentially what we do as geneticists is compare a bunch of dogs who don't have a condition with a bunch of dogs who do have the condition. And we try and look for something which is common to all the affected dogs, which is absent from all or most of the unaffected dogs. And if we do that in a very diverse population, like a bunch of humans from the UK or across the whole of the dog species, then it's really difficult to pull apart those patterns. But if we look within a modern dog breed, they tend to actually all be so similar that it's pretty easy to pick out the disease-causing mutation, which I've represented by these orange dots. Now, you may be able to do that in a more diverse species because those disease-causing mutations are likely still to be there, but it's an awful lot harder to pick the pattern out. And so you can see that there is a good reason to study genetics of a trait within a breed. And so that's what I did in the Labradors. And actually, I did something very simple in the first instance with the Labradors, because we know quite a lot about obesity biology. So I went off and I sequenced some genes that I thought might be good candidates for causing the obesity phenotype. And what I found when I contrasted the genetic makeup of a few lean dogs with a few fat dogs was that there was a mutation in a gene called POMC, which was pretty uncommon in the thin dogs and really quite common in the overweight ones. And that mutation is a deletion in a gene. We can be very confident that it would disrupt this gene POMC. And the reason I'd gone looking there in the first place is because it's part of this, which is a brain signaling pathway, which is quite central to regulating the body's food intake, energy expenditure, and ultimately body weight. And so we call this the leptin melanocortin signaling pathway. And this little cartoon over here is meant to be fat cells in the body. And fat cells produce this hormone leptin in greater amounts when there's more leptin around. And so leptin is released into the bloodstream and acts in the hypothalamus in the brain on nerve cells which produce this protein POMC. And when those POMC neurons are activated, they release downstream products of POMC. I've got a diagram of this later, called melanocyte stimulating hormones, with the net effect that when there's masses of fat, masses of leptin, then we see signaling through the pathway and a reduction in food intake and an increase in energy expenditure. Now, actually, what we know is that this pathway is most active and most important when it's disrupted. When there isn't very much leptin, because there isn't very much fat, the body sends out a starvation signal from this absence of leptin. And when there is disruption of signaling through this pathway, there is a massive increase in appetite and, to an extent, a decrease in energy expenditure, although that hasn't been very well proven in humans. It had been suggested in mice. And what we found in the Labradors is that there is a mutation in this gene POMC, and it disrupts signaling by melanocyte stimulating hormone at MC4R, melanocortin 4 receptors. And therefore, we would hypothesize that this mutation I found would cause precisely the kind of foodiness, food obsession that we see in the Labradors, and possibly that it might affect energy expenditure too. And so we went out to test that. Initially, I discovered this in just a handful of dogs. It was important to go and look in a bigger group. So in about 250 dogs, we looked at the effect of carrying 1 copy or 2 copies of this mutation in the gene POMC. And what we found is that in dogs who had 2 copies, they were on average about 4 kilograms heavier. Bear in mind your average Labrador is somewhere around a kind of 25 to 30 kilogram mark when they're at a healthy weight. So that's a pretty big effect. And we can express that not just as weight, which of course could be to do with how muscly or how tall a dog is, but also using something called a body condition score, which is a measure of adiposity, of fatness. And so the difference between dogs with no copies and 2 copies, all else being equal, is about a body condition score point, or about 10% extra body fat mass. Of course, we studied these guys because of their legendary foodiness. And so I spent quite a lot of time developing a questionnaire which put a kind of number on greed. And sure enough, and gratifyingly, owners reported that dogs carrying this mutation were more highly food motivated in the home environment. And so we published that work some years ago, but actually in the intervening years, I was quite interested in trying to drill down more specifically into the effect of this mutation on the neural pathways controlling appetite. And we've done that by trying to dissect out different aspects of feeding behavior. So first we looked at how much dogs would eat when given free access to eating essentially as much as they liked. So we gave them a series of meals, of cans of dog food, and saw how much they ate. And although all dogs in our studies, whether they had no copies or 1 copy of this mutation, ate an enormous amount, in the order of 2 kilograms of food at a sitting, actually there was no statistically significant difference between the 2 groups. Those dogs with this mutation did tend to eat more, but we couldn't, you know, it's not a huge difference. Where we did see something more interesting was when we used a different kind of test to try and get a handle on how hungry these dogs became. And so what you can see here is stills from an experiment where we looked at what we call motivational salience in response to a food cue. And so what you can see in this video is my colleague Gabby showing a hot dog sausage to Boody, who is a dog who is what we call wild type for this mutation. So she doesn't have a copy of the mutation. And you can see that she was pretty interested in the sausage, and she kind of pays attention to the box, but actually fairly quickly figured out that she couldn't get to the sausage and spent her time exploring this new environment. And that was reasonably typical of the dogs who came and took part in our study who didn't have a copy of the POMC mutation. Contrast that, if you will, with Ash here, who does have a copy of our POMC mutation. And Ash spent the entire of the 5 minutes of the video capture study really very excited and somewhat kind of frenetic in his pursuit of the sausage. I've turned the audio off this video because you kind of hear him squeaking away in the background. Now, intuitively, you can see that there is a difference in the behavior of those 2 dogs. And of course, we chose kind of good examples to show you for this kind of presentation. But importantly, these data have all been coded painstakingly, actually, frame by frame, to put a number on the kind of interaction with the cage versus with the little box versus exploring or kind of sitting down near owners or whatever. And what we can do when we graph that out and test it statistically is show that there is a clear difference in the way the dogs with the mutation respond to the sausage compared to those without the mutation. And because we did this in a standardized environment at a standardized time, about 3 hours after they'd had a meal, then we can say that the one big difference between those 2 groups of dogs is their genotype, and that we are seeing this difference because these dogs are more highly motivated by the food item, which is most likely because they are hungrier. Now, what about the energy expenditure? Now, I must admit, when I started doing this experiment, I really didn't expect to find anything. When people have looked at humans with sort of similar mutations, they've not, it's not been looked at in great depth, but there weren't striking differences in energy expenditure. So you'll notice that the dogs in these pictures are not Labradors, but flat-coated retrievers, because they're the only other breed that we found have this mutation. And as it happens, it's more common to find dogs with 2 copies of the mutation in the flat coat population. And so we recruited some of these lovely dogs and got them used at home to hanging out in our respiration chamber, which, as you can see, is a sort of modified air transport carrier actually for dogs. And what you can see in that bit of kit in the middle is a sealed chamber with very controlled airflow coming in at the bottom, at the front, and out at the back, that is pulling air through at a known rate and into a gas analyzer, which allows us to measure the amount of carbon dioxide that these dogs breathe out, and in turn to therefore calculate how much energy they use when they're at rest. So we wait until the dogs are fast asleep in the chamber and then measure their energy expenditure. And when we did that, we had a very striking finding. So I'm afraid I couldn't think of a really good way to show you this without facing up to a graph. But what we have on here is a graph that shows body weight on the x-axis horizontally at the bottom. And it kind of figures that we have that in there because if you're a heavier dog, you're going to use more energy just keeping your cells alive than if you've got fewer cells because you're a lighter dog. And that's why all of this, because it's metabolic rate on the y-axis going vertically, all these points tend to sort of slope up and right. But what we can see on here is that wild type dogs and those without the mutation, who are shown in white, tend to sit much higher up on the graph than those affected dogs with 2 copies of the mutation shown in black. And actually that difference is really striking. It says that when these dogs are lying in a cage at rest, doing absolutely nothing, the dogs with the mutation are burning off about 20% less energy than their wild type counterparts. That's a big difference. I've tried very hard to make these data go away because I didn't quite believe them to begin with, but I can't. It's real. And actually, it fits very neatly with what we know about this leptin-melanocortin signaling pathway. If it's broken, then the body is essentially receiving a physiological starvation signal, its response to which is eat as much as possible and conserve energy. And that's what we're seeing in this Labrador and flat-coated retriever mutation, which we have shown is a major modifier of weight and fatness. It affects both hunger and metabolic rate, and it's doing so in a fairly striking way. Now, it's important to say that actually only about 25% of pet Labradors carry this mutation, and yet people commonly say, it's not true, but they commonly say, oh, all Labradors are really foodie. And so there's plenty of other genetic stuff going on in Labradors, and I'm working on trying to find what they are, doing my best. But I think it's a good moment to reflect on those responses and what we can learn from Labradors that's relevant to our veterinary care of these patients, and also just to our understanding of obesity in general. And actually, this quote here is taken from a paper that reported the clinical features of human patients with POMC mutations. And here it said both patients were markedly hyperphagic, they ate a lot, as indicated by a score of 9 on a Likert hunger scale ranging from 0, no hunger, to 10, extreme hunger. So they felt hungry all the time. Psychological evaluations revealed this led to constant extreme dissatisfaction with their quality of life. Being hungry all the time is blimmin' horrible. And actually, I think this does present us with some quite real challenges in managing dogs with these mutations. Because when I was at vet school, we're taught about the 5 freedoms of animal welfare, 2 of which are freedom from hunger and freedom from disease, and obesity is certainly a disease. And so there's a real tension here between restricting food to keep dogs at a healthy weight and making them feel hungry all the time. And I think there are some very practical management interventions we can apply in affected dogs that I'm happy to answer questions on at the end, but which I think will improve clinical outcomes and welfare in these patients of mine. And from a practical point of view, it certainly means that owners of highly food motivated dogs, certainly carrying this mutation, are up against it when it comes to keeping their dogs slim. These dogs are opportunists. They are on the lookout for snacks at every possible opportunity, and they will work really quite hard, whether it's going off to find extra bits of food or just sitting there gazing adoringly and giving their owners the big brown eye treatment until they get a snack. And they really make an effort to get food, and it makes it much harder if you own a really foodie Labrador to keep it slim than it would be if you had a much fussier breed of dog. So all of that's kind of clinically important and relevant, but it wasn't what made me a magazine cover girl for the only time in my life. And the reason why this kind of high impact journal was interested was because of some quirks of POMC biology, which are particularly helpful when it comes to studying dogs as an animal model of human disease. So I mentioned earlier that POMC is quite a complicated gene in that it makes a protein, which I've indicated by the blue bar at the top here, which initially is a long molecule, but is cleaved, cut up by a number of enzymatic reactions into a whole bunch of different brain active fragments over a fairly complex processing effort. And the headline acts when it comes to obesity regulation or energy metabolism regulation in humans are these two, alpha and beta MSH. And they act at those melanocortin-4 receptors that were on our diagram. And we know that humans who lack either alpha or beta MSH are prone to overeating and becoming very overweight early in life. But actually the number of human beings who have been identified with beta MSH mutations is very small. And certainly there haven't been enough beta MSH mutation human beings to study systematically. So trying to work out what the difference between alpha and beta MSH and how each of them, these two very similar molecules, each are necessary and do something perhaps different when it comes to managing energy metabolism in the body. Studying that systematically has been quite difficult and impossible in humans. It's also been impossible in rodents, because through a quirk of evolution, rodents don't make beta MSH. They lack one of those cleavage sites, and rodents get on just fine with only alpha MSH and never produce beta MSH. Enter the Labrador, where in normal, and in all dogs for that matter, in normal dogs, they all produce both alpha and beta MSH. But in a quarter of pet Labradors and 2/3 of flat-coated retrievers, there is a mutation which, because of its nature, scrambles the whole of the end of this protein that's produced, and means that we can confidently say that while they have normal alpha MSH, they have no beta MSH. And so the smoking gun when it comes to causing Labrador obesity is this absence of beta MSH, and it gives us the chance to try and understand through those studies I've described how it is that beta MSH has its most important activity in the body. And so the Labrador example is quite extreme, really. It tells us about a really big mutation that kind of is very disruptive to the protein which the gene encodes. But it's worth pointing out that actually there's lots and lots of examples of less extreme, but equally clinically relevant disruptive genetic variants, which are relevant in humans too. And so all of the things in this table talk about genes that we kind of know what they do. So CCK, for instance, is a satiety hormone. So it's produced after a meal in response to protein in the gut, and signals to the brain that it's okay, we've got nutrition, and now you can have a sensation of satiety, fullness. And there are common variations in the gene CCK out there in the human population that, if you carry, are associated with extreme meal size. So you don't feel full until later. Similarly, there are variants in the leptin gene associated with extreme snacking behavior. I don't think that means eating your chocolate bar off the edge of a cliff or something. It's more about that sensation that actually I feel very familiar with. That I always have an eye on my next meal. I always know that it would be quite nice to have some food next. And I'm conscious and mindful of that, but I'm aware that it's actually quite different to my husband's sensation of food, and where he will eat well and likes to eat big meals, but he doesn't plan the next meal. He's not that bothered about snacking in between. And so these littler, more minor variants in genes are out there in the population, and each can have an incremental effect on our individual response to food, and therefore our tendency to overeat or not. And I think it's important to recognize that these are there, and that these altered or varied sensations and responses to food are real, they're hardwired, they're genetically driven, and it's nothing to do with people's moral compass or willpower. It means perhaps that those of us who are always thinking about food in between meals need to use a bit more willpower to overcome that and not to snack all the time. But even if we can do that most of the time, if we're up against a bigger drive, we're likely to slip more frequently. And I'm jolly well blaming a suspected leptin variant for my lockdown extra tire or two around my waist. And overall, what this does is point to that take-home message that genetically driven, hardwired differences in our biology and our responses to food underlie our neurological response to food, and therefore our behavior, our eating behavior and our food choices. And even if those changes are very subtle from one person to another, if they are in play all day, every day, for year after year, it will ultimately mean that some individuals are prone to sneakily gaining weight as time goes by. The other thing I think we can kind of quite objectively believe from studying the dogs, in a way that perhaps people are somewhat resistant to taking on board when it comes to thinking about human obesity and human biology, is this effect of the environment in which we find ourselves in terms of making food choices. And it's worth going back to that genetics and evolution story to think about the pre-domesticated dog, wolves, and how hard they have to work to get a decent, high-calorie, nutritious meal. Or even to think about the early domesticated dogs. This is a picture of a village dog in India who's probably living as domesticated dogs have for in the order of 30,000 years, albeit with plastic bags in the picture. But, you know, pottering around a human settlement, trying to find discarded scraps of food that are worth eating. Probably not going to get great tasty things there. And it makes sense to have a pleasure response in response to it, when they get high calorie, high energy food that will drive them to keep working hard to find those little morsels. And if you take a body that has evolved in that background and put it into a modern pet dog's world, where there is a plethora of food marketed towards pet owners, most of which talks about how delicious it is and how much your dog will like it, then it's hardly likely that they don't turn down whatever they're offered in their bowl. And then if all the hard work they have to do these days to get a little bit more food is to sit there and gaze adoringly at their owner, then, you know, it's hardly any surprise that they go to that effort. And to put that back in the Labrador retriever story, I mentioned earlier that the only other breed we found this in was flat-coated retrievers, and we can tell from the genetics that it was likely a variant that was inherited from a common ancestor, and it seems likely that that common ancestor was the St. John's water dog. And these were fishermen's dogs who went out on the boats in 16th century or 17th century Newfoundland, leapt into the icy waters, pulled in the fishing nets and worked incredibly hard for a living. And you'd have thought that in that context, a willingness to kind of snaffle any rotten fish head that was coming along would probably do you no harm, and certainly a layer of blubber, if you managed to develop one, under your skin would perhaps keep you warm as a dog rather than causing any trouble. We've also got a hint from the data in the Labradors that because this is more common in guide dogs, assistance dogs, maybe if you're a bit foodier, you're a bit more willing to work for humans. And that may have played in to Labrador's appeal as gun dogs down the centuries. And it's really only in the kind of modern day that we see Labradors affected by a debilitating obesity as a consequence of being so highly food motivated. So their environment has changed, not their genetics. And again, if we kind of turn our understanding of that hardwired biology in dogs to the human context, I think very few of us would pile on the pounds if all we had to eat was vegetable soup. We might be perfectly healthy. We probably would be very healthy, and it looks delicious. But I'm pretty confident that none of us would massively overeat to the point of obesity if presented with nothing but vegetable soup. Whereas if we were presented with cake, or as we are regularly offered sort of tempting high calorie, high sugar, high fat foods, then it's much easier to take on excess calories. And actually we get a real sense of reward and good feeling from eating something quite that delicious. And so that's a sort of binary choice of what you're offered. But realistically, we are offered delicious things every time we go to the supermarket. And most of us in the West, at least, are offered, have the ability to eat large volumes of energy-dense food whenever we want to. And it's no surprise, perhaps, that we therefore tend to make choices that promote obesity across the population. So just pulling all of that together, I'm interested in studying the biology of obesity, both to help humans and to help my veterinary patients as well. And we know that obesity causes a huge number of health problems in both species, and that genetics are very important in determining whether one individual is susceptible to becoming overweight, whereas others seem to be quite relatively resistant to developing obesity in this modern environment that we live in. I talked about the fact that dogs can be really good models for studying human disease because of their similarities in terms of the diseases they get and the environment in which they get them, but also because of their genetic differences and the way that dogs have been selectively bred down the centuries means that there are those genetic quirks that makes gene mapping quite tractable in my patients. We've studied that in the context of the retriever and his tendency to gain weight, and shown that a mutation in POMC has not only promotes food intake by making these dogs much more hungry between meals and more highly food motivated in their home, but also remarkably by having this big effect on energy expenditure too, which is I would stress unusual. You know, the take home message from this really is that in general genes affect our perception of food and the sensation of what it feels like to eat when we feel full and so on. And those differences between individuals' perception of food and how it makes us feel are real and biologically hardwired. And given that we evolved in a resource-poor environment and now find ourselves faced with plentiful, high-calorie food wherever we go, then it's perhaps no surprise that there's such a problem with obesity in both humans and dogs. Now, with any research talk, there's always thank yous to give. And the first and most important one in this talk is to say thank you to all the owners who've contributed to our study. We've gathered in the order of 2,000 dogs' DNA samples in the pursuit of this science, and people have been absolutely wonderful at contributing information and slobber from their dogs in order to allow us to do these studies. And then, of course, some owners have been particularly dedicated and amazing to volunteer to get their dogs used to the energy expenditure chamber, or to get them eating the right diet so that they can come and take part in our food experiments. And a lot of that work was done by Jodie, Marie and Gabby, who you can see in those pictures, who have been the researchers who've gathered a lot of the data as it went along. So I'm very happy to answer any questions now. You can find us on social media if you want to hear any more, and certainly do get in touch if you watch this on a recording and are interested in participating or finding out more.
Thank you very much for a really great talk. As you mentioned, we'll be taking questions. So if you do have any, please put them into the chat and I can read them out, for which you only have one. So Athea Maybury asks, why are both your obese figures female? Just asking. Great talk.
I don't think they are. Certainly the male-female figures I've got here are, I think, supposed to be a male and female differently. So it's important. And actually there are sex differences in the health consequences of being overweight in men and women. And there are differences in the nature of obesity and risk factors. And from a genetics point of view, it's been quite late in the day that people have kind of considered those and considered different populations separately. So it's something that we're all very aware of in the field.
So I was really interested to hear, especially the ancient origins and the bottleneck effect of the genetics. And so one thing you mentioned is that hunger reflex was likely developed and likely encouraged genetically in the past. So do you think maybe in 10,000 years, we may have the opposite problem, especially those who perhaps stay in a resource abundant environment for that long?
Yeah, it'd be fascinating to think, wouldn't it? I mean, certainly when I was first introduced to this field, the idea of a thrifty gene hypothesis, that our ancestors did evolve in a resource-poor environment and had very little selection pressure against becoming overweight, but did have a selection pressure against not eating enough, is a very compelling hypothesis. I think it's important to acknowledge that actually there are alternative genetic hypotheses that we may have ended up like this through a process called genetic drift, whereby stuff just kind of comes up in populations, and so not all of the obesity proneness that it comes through via genetics is necessarily down to that ancestral selection pressure, but it seems quite likely. Yes, if we hang around long enough, then I think there is likely to be a pressure, and particularly because, as well as the kind of well-publicized risks of diabetes and heart disease and stroke, which we tend to think of as middle-aged to older age conditions after reproducing, there are also impacts on fertility from being overweight through polycystic ovary syndrome and other mechanisms. And so, yes, there is potentially a fertility impact and therefore a selection impact from being overweight.
Interesting. One other thing, with the accessibility of genetic tests now getting easier and easier nowadays, do you think there'll be specific testing and perhaps treatment for obesity on a genetic level with patients?
Yes, so that's that's real and here with us today on several levels. And so my colleagues at the Institute of Metabolic Science, particularly Stephen O'Reilly and Sadaf Faruqi, have for many years studied patients with severe early onset forms of obesity and tried to determine what genes are responsible and how they exert their effect. And that's been hugely informative to the field in understanding this biology, but it's also been very important to those individuals who often have been unwell or sort of debilitated by their obesity from very early in life. And because of the nature of society being very judgmental about overweight people, have often had a fairly miserable kind of emotional consequence to that as well. And by providing those patients with a genetic diagnosis, it can be a hugely valuable boost. In addition, in some cases, they've also been able to move on and provide tailored specific treatment as a consequence. And so, for instance, we learned a lot about the biology of leptin from the study of patients who have an inability to produce leptin. And in those patients, Steve and Sadaf were amongst the first to treat with external, chemical, produced in the lab leptin. You can be injected and can reverse that early onset obesity and a plethora of other problems that come alongside it. So that's a huge and important thing. And actually recently it's been in the news about a drug called setmelanotide, which has been developed that acts on melanocortin-4 receptors. And so patients with mutations in POMC or patients with melanocortin-4 receptor mutations as well, and various other things have benefited from this very recent development in pharmacology that can treat their severe obesity. So drug testing has that very kind of precise effect there, but also on a more general basis, you can now, many of us, lots of people have had 23andMe or other similar sets of genome-wide testing, and those direct-to-consumer genetic tests are out there. And if you take them, just as they may tell you if you have particular cancer susceptibility genes, they will also tell you if you are particularly likely to be prone to obesity. And I think there was a bit of a worry in the field that if you told people they were obesity-prone because of their genes, then they'd just kind of give up, roll over, and say, well, I'll eat what I like then. Whereas actually, I think there is good evidence out there that it tends to either empower people to work harder to combat weight gain, or at least has a neutral effect, just allowing them to understand their tendency to overeat.
Yeah, and I think that really raises an interesting question for this field and many others. For, of course, if you have particular obesity or some other condition, you test for it. But what about for those who don't have any symptoms? Do you test for all these things all the time? Do you have any thoughts on that?
So would you do a genetic test for something that you don't have already? Mm-hmm. I mean, I don't know that there'd be much clinical reason to do that, would there? But certainly things like 23andMe allow people to get testing for something that they don't have already. That has ethical implications because you tell someone that they might get a problem in the future. With obesity, I think that's less of a problem because people aren't quite so afraid of it, and they understand that ultimately we have control on what goes in our mouths, although it can be a lot greater effort for some people to exert that control than others. That's a bigger deal with cancer testing, for instance. Should you, if the kind of classic thing is that someone gets bought a genetic profile as a kind of quirky birthday present and then discovers that they are prone to be a particular kind of cancer, and that can be quite a frightening experience, but perhaps less relevant with obesity. It's been something that we've thought about hard in the dogs as well, because having identified this in the Labradors, we know that it's real, that it has an effect to have this mutation, but that ultimately lots of owners of dogs carrying the mutation can control their dog's exercise and food and keep them at a brilliant healthy weight with a good quality of life. And so should you test for that? Would it make people work harder to keep their dogs slim, or might it just mean that they give up? And we have dealt with that by offering the test, but giving really quite intensive support literature that goes out with the results, so that people can have support and confidence that they know how to control their dog's weight if they carry this mutation.
Yeah, absolutely. Well, I don't see any more questions, and we're coming up on 3:30. So I'd like to thank you again, Eleanor, for a really great talk and discussion.
Thank
and
you.
Since this is the last talk, I have to thank all the speakers and especially all the viewers, both current and hopefully future, who will be watching this. And if you are interested or missed out on any of the talks, this video should be available in the future on this particular channel. And so with that, hopefully next year, we will be able to do this in person and see you all again in the probably rainy Cambridge streets. And so until then, I'll say goodbye from Cambridge and particularly PDN, and hope you all are staying safe. Take care, everybody.